He is with a vibrant mind and vibrant ideas and always thinking of something new. So that made him to achieve his goal in US, and now he is heading an institution center there. So Dr. Muralidhar Hegde has published large number of papers and a large number of collaborations, including our students. Any one of you have some ideas, we'll be happy to support you. Also send you for short visits or long visits, whatever you want. So we have the opportunities to work. So big, but important aspect is his dream is to identify the reason why movement disorders start and why a cell dies in that process. So we're going to listen to his lecture in a broader perspective. Thank you very much.
Hello, sir. Hello. Sir, ಇದು ಹೋಸ್ಟ್ ಮಾತಾಡೋದು ಕೇಳ್ತಾ ಇಲ್ಲ ಹೋಸ್ಟ್ ಅಲ್ಲಿ. ಹಲೋ. ಹಾ ಶೇರ್ ಮಾಡ್ಲಾ ಸರ್? ಸ್ಲೈಡ್ ಶೇರ್ ಮಾಡ್ಲಾ ಸರ್? ಹಲೋ. ಶೇರ್ ಮಾಡ್ಲಿಕ್ಕೆ ಅಲ್ಲ ಶೇರ್ ಮಾಡ್ಲಿಕ್ಕೆ ಅಲೋ ಮಾಡ್ತಾ ಇಲ್ಲ ಸರ್. ಅದು ಹೋಸ್ಟ್ ಅಷ್ಟು ಗಿವ್ ಪರ್ಮಿಷನ್ ಅಂತ ಬರುತ್ತೆ ಬಸ್. ಹಾ ಈಗ ಬರ್ತಾ ಇದೆ ಮೋಸ್ಟ್. ಮಾಡಿ ಒಕ್ಕ ನಿಮಿಷ ಸಂತೋಷ. ಕಾಣಿಸ್ತಾ ಇದೆಯಾ ಸರ್ ಈಗ? ಕಾಣ್ತದ ಬಿಡ್ಲಿ, ಸ್ಟಾರ್ಟ್ ಮಾಡು. Good morning, everyone, and it's actually night from USA, Houston, Texas. So my name is Muralidhar Hegde, and I am professor of neurosurgery at Houston Methodist Research Institute, which is an affiliate institute of Cornell University in Houston. And first of all, I would like to thank the organizers, particularly Dr. Rao, for inviting me to share some of our recent discoveries on DNA damage and repair aspects in neurodegenerative diseases. Particularly in the last few months, we found a unique and very interesting link between a new class of proteins called RNA DNA binding proteins, which were identified in other neurodegenerative diseases. But we found a unique link of their involvement in dementia-associated diseases, including Alzheimer's, and how the DNA repair is significantly compromised because of these proteins playing a role in addition to classical Alzheimer's disease pathology in these diseases. So I would like to give a brief outline of what I'm going to present. So in the first half of my work presentation, I would like to share some of the new discoveries we made on linking genome repair defects in neurodegenerative diseases, including AD. So, as you know, for a long time, for about 2 to 3 decades, genome damage has been shown to or involve have been associated with neurodegenerative diseases. But more recently, we and others have been trying to investigate how this DNA damage accumulates in diseases. Is it because of damage induction or whether the repair is compromised? It turns out that you know there is a continuous induction of damage, of course, which occurs in all organisms. But there is also efficient repair of those damage. And if the repair is compromised, as it occurs in these diseases for various reasons, the damage accumulation aggravates. And we have identified some unique mechanisms of these DNA repair defects and how the damage and damage signaling contributes to these neurodegenerative pathways. And based on these new mechanisms, we have also identified some interesting ways to, reduce or prevent these repair-induced neurodegenerative pathways, which I am going to present today. As you know, there are more than 200 or 250 neurodegenerative diseases known in humans. Although the common ones are Alzheimer's, Parkinson's, and motor neuron diseases and so on. But among these large classification of diseases, there is about 10 to 20 different subsets based on the unique pathology and unique causative features, sporadic or familial, which may include several dozen genes. So each each group of patients may have a unique pathology, a dominating pathology. So if you consider this as a complex subset of diseases rather than a single disease. So there are several diseases known in humans, although sometimes the symptoms appear to be similar. The underlying pathology, which is very difficult to identify in live human patients, till you get the postmortem tissue to state to pinpoint what subset was dominant in those patients. So it's important to mention that these are progressive diseases. The neuronal degeneration continues to occur, and although there are some therapeutic principles available to slow down the disease, but the ways to prevent those neuronal cell death is not there. We are still not there. So the cells continue to degenerate, and ultimately the therapies do not work. So and most of them are sporadic without any known cause, and about 5 to 10% are inherited. So when I was working with Jagannath Rao about 20 years ago as a PhD student, we were looking at what are the common causative features, what are the universal causative contributing factors to diseases, and it turns out that predominantly metal toxicity and oxidative stress, mostly redox metals and heavy metals. The combination of metals could be different. In some cases, it's occupational exposure, or in some cases, it's just reorganization of that metal homeostasis or homeostatic imbalance, which are associated with diseases. And protein misfolding and aggregation, which has been the focus for a long time, occurs in all these diseases. It could be amyloid beta, tau, alpha-synuclein, and Parkinson's. And as I mentioned, a new class of proteins called RNA binding proteins. I mentioned TDP43. There are other related proteins which were first implicated in amyotrophic lateral sclerosis, which is a motor neuron disease, but later on, in majority of the dementia-associated diseases, these RNA binding proteins, appear to be significant contributing factors at some stage of the disease. A third common feature is accumulation of DNA damage, which is, of course, our main interest in our lab. And what's important here is, although these common pathologies or etiological factors appear during disease progression or initiation to a distinct, unique extent, but what are the relative contribution or crosstalk among these etiologies? This is not very clear. And there is a lack of understanding of specific repair defects or the connection among these factors, which is a roadblock for exploring mechanism guided therapeutic concepts, and which is the main issue we are trying to address. And the other issue is, as you know, in Alzheimer's disease, memory-related cells degenerate, and in Parkinson's, it's substantia nigra or midbrain regions, which are responsible for dopamine production or storage. They degenerate. So why and how these unique sets of neurons are affected in a particular disease? Why they are vulnerable for these etiological factors, including DNA damage and repair defects? It's an extremely important question, and it has remained as a question for a long time, but we have some interesting clues in the last few years, and we are very close to understanding, to some extent at least, as the disease progresses, why these particular group of neurons in these diseases get affected. And it has always been a challenge to study these diseases, particularly in humans, because we don't have the appropriate technologies to look at the cells as the disease progresses. There are imaging techniques available for tau and A-beta, but for other proteins like synuclein and GDP43 or other RNA binding proteins, we still do not have diagnostic imaging facilities available. So animal models mimic only partial disease phenomenon. Depending on, most of the time they're very aggressive because you have to represent the disease in a short period of time, and they show the symptoms, but when you want to explore them for therapeutics, because they are very aggressive, so they don't turn out to be that effective. So we only have a clear picture of this disease pathways in the postmortem tissue after the patient dies. That has been an extremely difficult challenge in these diseases, and we have made some strides. We and other groups all over the world have made some strides in making use of trying to overcome these limitations of non-availability of techniques to detect these diseases, as well as non-availability of very closely disease mimicking organisms, by using a complementary approach, in the sense that using patient-derived cells, patient-derived mutant cells for the disease isolated from disease patients, and animal models, as well as patient tissue. Using this combination, arrive at some consensus about the disease models. So, in addition to these complexities, brain also is a network of many cells. Neurons are the central players, but neurons are supported by astrocytes, interneurons, glia, and several other cells. And of course, neurons have to die for the disease to occur, but neurons may die as the disease initiates. In neurons, neuron may be the initial culprit, or astrocyte could be the culprit, which ultimately affects the neurons. And these multiple cell types differentially or distinctly contributes to the disease phenomenon. That adds another layer of complexity. And the other thing is, many of you may be aware that neurons are the longest living cells in our body. So most of the neurons what we have, we develop within 3 to 5 years of our age, and then they are trained for various functions. And after about 20 to 30 years, they slowly start aging. Am I audible? Is there an issue? Okay, let me continue. So, because these are very minimal. कैसा है जरा सर? I'll try to speak a little bit closer so it's not like that. So, because neurons are long lasting, they tend to accumulate various toxicants. They tend to accumulate various pathway defects, and that's what which occurs in this age-associated diseases. So sometimes the when the disease symptoms appear, the build-up must have occurred about 20, 30 years before. So it's very difficult to pinpoint what caused the disease unless there is a unique mutation in a particular protein in some, particular percent of familial diseases. And another thing is, neurons, brain is highly active. Even when you are sleeping, brain is the organ which is always functioning. And in fact, if you measure the functionality of body cells or body organs with our breathing or oxygen consumption, about 70 to 80% of the oxygen we breathe goes to the brain. So the oxygen we breathe is involved in, it's required for energy production and various metabolic activities. And it also produces the byproducts of reactive oxygen species. That's the reason brain cells are continuously exposed to these redox agents, and that's one of the sources of this DNA damage. But they also, because this occurs continuously as a physiological process, brain cells have also evolved effective pathways to counter these byproducts or side products. And that's how they are long lasting and they live as long as we live. But any changes in those pathways then tend to accumulate mutations and and defaults in the system. So coming to the DNA damage, as I already mentioned, in aerobic organisms, which is oxygen for the functioning, so multiple kinds of DNA damage occurs in every cell. It continuously occurs, and it could involve base modifications by oxidants. It could involve single strand breaks, double strand breaks, base adapts, and so on. And we have evolved multiple repair pathways for each of this damage. That's how we repair and maintain the DNA integrity. So as per an estimate which was done by Thomas Lindahl, for which he got a Nobel Prize a few years back, about 10 to the power of 6 just base damages and 10 to the power of 5 single strand breaks occur in a cellular genome, in a single cellular genome every day. So if you think about this huge level of damage and the cells still maintain the DNA integrity, you can imagine the effective or proficient repair pathways cells have developed. And this is one example of the DNA damage, double strand breaks, which is lethal because both the strands of the DNA are broken, the DNA gets separated and transcribe or replicate and so on. So there are 3 pathways for these cells, and many of these pathways work based on cell cycle or cellular stress conditions and so on. And if one pathway is defective, the backup pathway gets employed. So we have those effective pathways. However, in some of these neurodegenerative diseases, we still accumulate this DNA damage. And we were wondering, with availability of 3 backup pathways, 3 pathways, how we still accumulate the DNA damage. And we have found some unique pathway defects and mechanisms that I'm going to present in today's presentation. And that also provided a very unique connection to Alzheimer's disease and dementia. And DNA repair in humans is highly complex. In a single cell organism like bacteria, it is very simple with 4 or 5 steps like chemical reaction. You can kind of reproduce the basic reaction in a test tube. However, in a human cell, it's much more complex because the DNA, broken damaged DNA, is held by a huge complex of proteins, about 3 dozen proteins, and they are post-translationally modified, and there are unique proteins in dividing cells, in transcribing cells, and in heterochromatin, euchromatin, and so on. And we have characterized, we are one of the pioneering labs when I started working as a postdoc in Mitra lab several years ago. Pioneering lab to identify mechanism of this basic repair pathways, particularly complexities of DNA repair in mammalian cells and in human cells. And we are looking at several pathways, but uniquely, one new class of proteins we found that got involved in these diseases is in the repair pathways is RNA binding proteins. We were looking at HNR and PU, which is primarily involved in RNA metabolism like transcription, translation, and so on. But we found that the protein plays a key regulatory role in DNA repair. When there are multiple damages, instead of single damage, if you hit the DNA with a radiation, for example, or a chemical, then you induce multiple damages in a close proximity. That could be double strand breaks, that could be base damages, and so on. Now, multiple pathways have to operate in a very close region. And we found that these pathways are now temporarily regulated. Like most lethal form of damage is repaired first. That means those pathways repairing those lethal form of devastating breaks are employed first, and then followed by base damage. And this kind of switch, we call it as a molecular switch between those repair pathways, is regulated by HNR and PU through its phosphorylation and dephosphorylation phenomenon. And I'm not going into the details of this work, and we published it several years ago. But the reason I'm mentioning this is when we are looking at these new class of proteins in fundamental aspects of DNA repair, two groups, particularly Virginia Lee and another group in St. Louis, Paul Taylor, they were looking at these RNA binding proteins in some neurodegenerative diseases and found two other RNA binding proteins belonging to the same HNRNP family called TDP43 and FUS were implicated in amyotrophic lateral sclerosis and frontotemporal dementia. These are two motor neuron diseases, one affecting the spinal cord, the other affecting both motor cortex as well as hippocampus. So you have both motor neuronal defects, motor defects, and dementia. So these diseases, about 95% of these diseases, these patients had the aggregates of TDP43 like amyloid beta aggregates and Alzheimer's disease. These proteins, which are extremely important, if you remove this protein, the cells die. This is an essential protein. But the protein clogs, like it kind of clogs in the cell, does not get clear, and tries to build up as a junk in the cell. And that was shown in 2006. At the same time, we were looking at HNRNP family proteins in DNA repair. And most interestingly, Paul Taylor from St. Louis, we're looking at what are the interacting partners of these proteins. Those are the initial studies that were carried out. And some of those proteins identified in a large proteomic study were DNA repair proteins like COO. And that kind of triggered our interest because we were looking at these RNA binding proteins in DNA repair. And now some of these related proteins were implicated in neurodegenerative diseases, which also show a huge amount of DNA repair defect and damage accumulation. We were wondering whether there is any DNA direct connection in these RNA binding protein associated disease patients. I should also mention that before going to our discovery, that ever since TDP43 and FUS were implicated in ALS, subsequent studies showed about 40% of all neurodegenerative diseases have these protein defects. And more recently, studies showed about 70% of Alzheimer's disease patients have this pathology, and patients who have this pathology are more aggressive and resistant to therapy. So it's clear that these proteins play an extremely important role in Alzheimer's disease as well as other neurodegenerative diseases, particularly during disease progression. So we started looking at whether these proteins play a role in DNA repair. And we looked at the interaction with DNA repair proteins, and I'm just showing the positive data here. So we used neurons, the patient iPSC cells and patient iPSC cells converted into neurons in the test tube or in a petri dish. You can see 90-95% of cells are converted into motor neurons here. And take a laser. This part. So 90 to 90% cells are converted to neurons, and we found that in these cells, the TDP43 interacts with many of the DNA repair proteins like 53bp1, Q, XRCC4, gamma activation. These are classical DNA damage markers and repair factors. But interestingly, the basal level of interaction dramatically increases if you induce DNA damage by radiation or chemotherapeutic drugs, the chemo drugs which induce DNA damage. That shows that the interaction of this TDP43 and we later on we show for a related protein called first with DNA repair proteins is damage dependent. And we did that with an assay called proximately ligation assay where you can in cell you can identify this interaction and you can see when there is a break in a small movie clip you can see when there is a break we use primary antibodies attached to two oligos and these oligos are complementary sequences. So if the two proteins are in close proximity then these oligos bind together to form a circle and then that can be amplified by rolling circle mechanism. And then using a fluorophore to a complementary oligo, you can detect those interactions. So basically, in this video, each spot indicates one interaction. So this is an extremely efficient system, complementary to immunoprecipitation, which we also show here, supports the proximity ligation assay. So then we followed it with another method. We use multiple methods to consolidate this idea because this is the first time nobody has shown for the DNA repair role or association of these proteins with DNA repair. So we wanted to establish this as solidly as possible. So we used a laser ablation. We used a laser. So we used a laser to induce DNA damage in live cells. You can see when we hit the laser, these cells were expressing fluorescent TDP43 like JP43. As soon as you hit the laser, the TDP43 lines up on the laser track. And this is as fast as the classical DNA repair protein Q. So which suggests that TDP43 goes to the site of DNA damage within 1 minute of inducing that DNA damage and it remains there till the damage is repaired. So this also suggests that along with its interaction with repair proteins, when you induce damage in live cells, TDP43 actually associates with damage. And then we do what is the role of TDP43 on DNA repair? So we knock down TDP43 by siRNA, we knock down TDP43 by CRISPR-Cas9 technique, and with multiple sRNA and so on. Here I show a conditional CRISPR-Cas9 because if you completely knock out TDP43, cells die within 4 to 5 days. And because it's apoptotic death, the cell death associated DNA damage interferes with any DNA damage quantitation. So we did a conditional downregulation of TDP43 using a conditional or inducible CRISPR-Cas9 gene editing technology. And you can see here you can gradually knock down TDP43 with this method. And a small amount of decrease results in a huge increase in phosphorythrium, gamma activates, and so on before the cells start dying. And this was confirmed by Comet assay as well. And you can see as TDP43 decreases, there is increase in DNA damage. As TDP43 decreases, there's a dose-dependent increase in DNA damage. And we then confirmed that most of this damage are double-strand breaks by using a combination of neutral and alkaline Comet assay. Because as you know, alkaline Comet assay represents single-strand breaks as well as double-strand breaks. But neutral Comet assay kind of identifies exclusive double-strand breaks. But we found similar level of strand breaks in both alkaline and neutral Comet assay, which suggests that most of the damage what we observed were double-strand breaks. So now we know that TDP43 associates with double-strand breaks or DNA strand breaks and associates with the repair factors. And if you knock down TDP43, there is accumulation of DNA damage. So what does TDP43 do? So what we did here is we induced DNA strand break using a restriction enzyme based cutting of DNA called ISCA1. We introduced this plasmid and then used the restriction enzyme to cut the DNA. And because this plasmid has a GFP reporter, if the damage is repaired, then GFP gets expressed. You can see here with no ISCA1, there's a significant increase in GFP, and once you induce ISCA1, there's an increase in DNA damage, DNA repair, GFP expression. But if you downregulate TD43, the expression decreases, which suggests that TD43 actually affects repair of these double-strand breaks. Then we use various animal models here. We use C. elegans lacking a functional TDP43, which not only accumulates DNA damage, but also affects repair of those DNAs. And then we looked at postmortem tissue from these patients having TD43 pathology. We also show accumulation of DNA damage markers like gamma-2X and Strand-Breaks weight analysis. And the same patients show in the huge amount of DNA damage markers compared to control patients, which is more or less cleaner. So then we looked at what is the mechanism of a role of TDP43 in DNA repair. And I'm just going to present a snapshot or a representative data. We found that TDP43 is actually required for ligating the DNA strand breaks. If you don't have TDP43, the ligation activity is affected. So it looks like TDP43 traps this ligation complex, XRCC4 ligase 4, which is required for DNA strand breaks ligation in human cells. And the TD43's mislocalization in the cytosol as a junk traps this complex, so it's not available to repair the DNA damage. And in healthy cells, TD43 actually helps to recruit this complex at the damaged cells, which is probably in all likelihood. It looks like it appears that TD43 kind of acts as a glue to fix the DNA strand, but broken DNA on one plane, so that the repair ligation reaction can proceed. And this work was, this was the first evidence to show that TDP43 is linked to DNA diverse strand break repair defects, and we published that in PNS in collaboration with Jagannath Rao and Paul Taylor and others. And in familial patients having TD43 pathology, we found that the mutant actually kind of sequesters the XRCC4 ligase for DNA repair proteins and make them not available to repair the DNA, and acts slightly differently in sporadic cases. And when we publish this follow-up work in human molecular genetics. And as I mentioned, TDP43 pathology involves its kind of sequestration into non-functional solid form, particularly in the cytosol. In normal cells, it's present in the nucleus. So there is a nuclear clearance or mislocalization of the protein. So we were wondering the repair defect, what we observed, whether it's because of the toxicity of the junk protein or its loss of its function. To test that, we engineered TDP43 vector without nuclear localization sequence and nuclear export sequence, so that we can distinctly or specifically knock down protein in the nucleus while it still expresses in the cytosol, and vice versa. So its nuclear clearance led to DNA accumulation, but not the cytosol specific downregulation, which suggests that TDP43's functional loss is responsible for this DNA damage defect. And we can clearly see here with the immunofluorescence, the loss of protein from the nucleus leads to damage markers in the nucleus. You can see no protein in the nucleus, then there is DNA damage. So based on this, we developed our own mouse model. At this stage, there are some mouse models like transgenic where you overexpress human TD43 in mice and so on, so which were not really representative of human TD43 pathology. But we developed our own mouse model where we can conditionally knock down the protein in the nucleus in an age-dependent fashion. And this we developed using a CRISPR-Cas9 technology, and we basically mutated the nuclear localization sequence containing exon and put it in the next intron in reverse orientation. So in a normal mice with this or this transgenic mice, it still expresses the normal TDP43 because the mutant is in the reverse orientation. But when we induce with a CRE, then the wild type analysis deleted and the mutant comes in the right orientation, and then the mutant start expressing mislocalized or non-functional problematic TDP43. You can see here it starts accumulating. And this mice, which we believe that is really ideal model to study TD43 pathology, or one of the better models to study TD43 pathology, shows the damage accumulation as it occurs in patients. You can see the several mice show high level of DNA damage compared to natural controls, and it also shows several behavioral defects, including motor and uh, cognitive defects. And the mice is very flexible because you can specifically knock down the protein or knock down knock in the mutant in hippocampus, in spinal cord, or in whole brain, or in different cells like neuron specific, astrocyte specific, using specific free expression mice, which is already available commercially. So, in summary of this part of the work, we were the first group to show that TDP43 plays a role in DNA double-strand repair, and its pathology in neurodegenerative diseases affects the DNA repair and leading to damage accumulation and damage signaling, which contributes to the disease. So, as I mentioned, these diseases are not single diseases. There are half a dozen different proteins, including related proteins like FUS, involved in these different diseases. We looked at one of the related proteins first, and we published in Nature Communication at the same time. We found that the FUS plays a role in DNA repair, but not in double-strand break repair like TD43, but in single-strand break repair. So it looks like these proteins have unique or distinct repair role in different pathways. And here again we use normal and patient-derived cells to show that FUS is involved in single-strand break repair pathways and associates with single-strand break repair proteins like XRCC1 and ligase 3, unlike TDP43, which associates with XRCC4 and ligase 4. And then in this case, we generated, we got, developed cells from the patients containing the FUS mutations, which showed the damage accumulation as well as repair defect. And to attribute it to the particular FUS mutation, we corrected those mutations by CRISPR-Cas9 knock-in mutation correction. And once you correct the mutation, the repair activities were recovered, which suggests that the mutations were actually involved in repair defect. And what does it do? So, let me go back to the end. So here we show that first wild type cells, when you hit the laser damage, accumulate XRCC1 ligase 3 immediately to repair the damage, but first mutant cells don't accumulate those damage. You can see it's very slow and the intensity is very low. So basically, first again, like TD43 affects those strand break associated ligases, or in this case it's XRCC1 and ligase 3. And we have I'm not going to show all the data for this work, but basically here we show that FUS has a distinct role in DNA strand break repair, and its mutations affect recruitment of those ligases and leading to accumulation of those oxidative DNA damage. And as a follow-up of this, XRCC1 and ligase 3 is an important protein in nucleus, but more importantly, it's the only ligase in mitochondria. Mitochondria has its own DNA, it replicates, and it also has to be repaired because damage is much more than the nuclear DNA. And there's only one ligase in mitochondria. As I mentioned, DNA strand breaks have 3 repair pathways in the nucleus. There are backup pathways, but in mitochondria, there's only ligase 3. So we thought, or we kind of conceptualized that if FUS is playing a role in mitochondria, nuclear DNA repair for ligase 3, its impact in mitochondria could be much more significant, and that's exactly what we observed. And this paper is now under review in Nature Communication. Like Manohar Kodavati, who is very close to your place in Rajahmundry. He's actually he's been working in our lab for some time now. He's doing a great job, but he is now visiting India for a vacation. I think he visited KL yesterday, which I learned from Jagannath Rao. So what he shows here is first localizes to, there are 2 distinct things. So first is required for mitochondrial DNA repair, and now what's its implications in neurodegenerative diseases. So Manohar showed here by both Western and immunofluorescence that first endogenous FUS localizes to mitochondria, and the mutant FUS, pathological FUS, goes more to mitochondria. And then he used these patient-derived cells as well as knockdown cells to understand what happens to mitochondria using seahorse, the mitochondrial energy metabolism. Several parameters were significantly affected when the first is mutated or knocked down, which suggests that first pathology actually affects mitochondrial functioning and activity. And here we show that first actually helps, again, as in the nucleus, helps with recruitment of mitochondrial ligase 3 to the nucleus, and this is where Chippas Emanuel shows that if you don't have first, the recruitment of ligase 3 to mitochondrial damage sites is affected. It affects the mitochondrial DNA damage repair. You can see without first, the repair is significantly lower, and it's also its interaction with ligase 3, mitochondrial ligase 3 exercise is significantly affected. So what we can do to recover that, because there is only one ligase 3 in mitochondria. So we wanted to introduce another ligase. Initially we thought of introducing a bacterial ligase, but then we used the same mammalian human ligase to avoid immune reaction and so on. The ligase 1, which expresses in dividing cells but does not express in neurons, because neurons don't divide. So we added a mitochondrial localization signal by genetic engineering to the ligase 1 construct, and expressed the ligase in the mitochondria in a targeted fashion. The targeted expression of ligase 1 rescued mitochondrial DNA damage, repaired defects, as well as restored mitochondrial functionality, even under extreme first pathology. This suggests that the ligase is actually contributing to the first associated neurodegenerative disease mechanism, and expression of ligase 1 could restore this functionality. This we did in patient-derived cells, but now we are trying to do in animal models and take it to clinical trials. And that's what represented in this phenomenon, where first plays a role in mitochondrial DNA repair, and during first pathology, mitochondrial repair is completely compromised because of the ligation defect, and introducing the ligase in mitochondria restores this functionality. So in summary of this part, we found that these two RNA binding proteins play a role in distinct repair pathways, and because of that, when they become junk, when they start at nucleus sequestering, there is a substantial repair defect which leads to damage accumulation and damage signaling, leading to neuroinflammation, senescence, and ultimately contributing to disease phenomenon. So can we use this ligase complementation to prevent TDP43 and first neurotoxicity? In cells, it was very positive for TDP43 for first percent TDP43, but we kind of hit a roadblock with TDP43 with ligase complementation. The reason was, although ligase was dysfunctional, when you induce ligase 1 in TDP43 cells were not repairing those damage. The damage was still stuck in this devastating base. We found that the backup pathways in the nucleus which are available are not employed when TDP43 pathology affects a particular DNA repair pathway, like NHEJ is affected, but the backup pathways are not employed. So we looked at the mechanism of this, and we found that a protein called BRCA1 is critical for this pathway switch. So because the backup pathways require more end resection and more energy, to prevent that, the preferential pathway is always directed. So if the preferential pathway is not working, then BRCA1 goes to the side, kind of kicks out the NHEJ preferential factor 53-1 and drives the damage towards HR and MMEJ, homology mediated repair pathways in normal cells. But we found that during TDP43 pathology, that's not happening. So the preferential pathway is not repaired, but those proteins are not removed from the site. The pathway switch is affected. So when we dig deep, we found that TDP43 as an RNA binding protein affects BRCA1 splicing. So if you don't have TDP43, BRCA1 is misspliced, and misspliced BRCA1 is mostly in the cytosol along with TDP43. It also sequesters. And that was interesting, that pathway switch factor is also affected by TDP43. And then if you look at Alzheimer's patients, mostly or dementia patients, The BRCA1 exactly behaves that way, as we showed in TD43 pathology mediated cell lines. So in Alzheimer's patient tissue, or in animal models, you see that BRCA1 defect. So it shows that TDP43 probably mediating BRCA1 defect, and those DNA repair pathways are completely compromised because of this pathway switch defect. And again, we show in animal models and other systems that TDP43 mislocalization induces BRCA1 clearance and increases DNA damage. That contributes to those substantial repair defect where backup pathways are not employed. And we also showed that in our TDP43 mice model where BRCA1 is defective, and then mostly those neural stem cells or in the dentate gyrus and hippocampus or neocortex, where a small pool of neural stem cells are there which contributes to and these are migrating cells, replenishing some of the memory related cells, and they are also required for memory cognitive plasticity to learn new memory and so on. These cells are affected because BRCA1 is a breast cancer associated protein involved in replication associated repair. Presumably, this plays a role on this small pool of dividing cells in the brain and affect those cognitive plasticity, thus contributing to memory. So, what can we do to address this? One way was to overexpress BRCA1, but TD43 pathology, which is not fixed, continue to misplace it. And BRCA1 is a huge protein, very difficult to overexpress. But we're trying to identify ways where you can now forcefully kick out those NHJ factors and kind of engineer the pathway switch in the absence of BRCA1. It turns out that this 53BP1, which is normally kicked out by BRCA1 and in one pathway is defective, has a DNA binding domain called pseudo domain, and there are in several other cancer studies, people have identified a small peptide called I53, which actually interferes with that 53 plus DNA binding. In fact, in a BSA knockout mice, similar repair defect is observed, but if you add that I53 or don't regulate 53BP1, then in spite of DNA damage, cells repair the damage using backup pathways. So we need exactly similar mechanism in neurodegenerative diseases because something similar occurs. So we introduced that I53 and found that now it kicks out the pathway switch factor which was stuck and restores the cell viability as well as DNA damage response by using the backup pathways. This is like facilitating the cells to repair its own damage using backup pathways rather than introducing a new pathway or new protein. And this is very promising. And now if you complement it with ligase 1 therapy, which we were originally doing, by kind of restoring that pathway switch and then complement a ligase, that kind of leads to a very proficient recovery of the DNA damage in the cell lines. And we call this a two-hist therapeutic strategy because it's ligase one complementation after restoring that pathway switch defect in dementia-associated conditions. This is not only in Alzheimer's disease, but in several dementia predisposing conditions like micro bleeding or brain trauma associated memory defects, or during impact injury in sports persons, which leads to some cognitive plasticity defects. So it appears that this is what exactly occurring, which kind of continues to accumulate and leads to permanent cognitive damage. So TDP targeting TDP43 through this DNA repair pathways may be one of the answers to this. And now we have this targeted therapy by 53 inhibition using I53 as well as with ligase 1 to develop this. And we are now using this approach. And I53 is a very small peptide, can be introduced without gene therapy using a peptide delivery. And we are trying to do that in our mice model as we speak. So if it's promising, it will go to clinical trials. So we have several other projects on Parkinson's and stroke where we also showed how DNA damage response signaling actually contributes to the neuronal side. Although these were in other conditions, that knowledge we gained was important to understand how the DNA damage actually is playing a role in progressive degeneration of neurons. And I'm not going to present that to keep it simple and straightforward. So basically, we not only identified distinct DNA repair pathway defect mechanisms, but identified a unique connection to Alzheimer's disease through that BRCA1 defect and its crosstalk with TDP43. I would like to acknowledge, people who have contributed to this work. I presented Manohar's work, which is under revision now, and Joy's work on TDP43, Erica Guerrero, who worked with me, and Jagannath Rao, originally from Panama, as a graduate student, contributed to some of the TDP43 studies, and several others like Wellman, Vincent Provosek is working on TDP43 and mismatch repair, and several others contributed to different aspects of these functions. And I also have a picture of Janaki Ramaya from KL who visited our lab for a short time, but did an amazing job in those short 3 months. We were really impressed as a faculty. He came over here and straight away got into wet lab work and identified some of the TDP43 associated repair defects in GBM and brain cancers. The kind of new phenomenon in those cells, those conditions where TDP43 is associated. So finally, I would like to acknowledge several funding from, particularly from National Institute of Health and various other organizations, and want to thank Jagannath Rao, my mentor, and he continues to be my mentor and collaborator in several of the studies. We continue to collaborate, and it's a guiding force behind many of the studies. And our lab members, several collaborators from all over the world. And thank you very much for listening, and I'll be happy to take any questions. Hello, sir.
Any question? Any question? No questions. We congratulate Dr. Murli.
Thank you, sir.
A very good morning to one and all, and welcome back to the fifth day of 5-day workshop of AI and ML. And I would like to thank Muralidhar Hegde, Professor Muralidhar Hegde, and we are very much thankful to, first of all, his mentor who is here rightly available with us, Professor Jagannath Rao sir, our Pro-Chancellor. Sir, thank you so much for providing such a wonderful opportunity to all the participants. And sir, participants might have observed once sir was saying about Professor Janaki Ram sir, who is from the Department of Biotechnology. He visited for 3 months and he did the lab research over there. And so there are a number of collaborations which are available, and we are very proud to say that our Pro-Chancellor has innumerable collaborations with all over the world. And we really, with a round of applause, let me ask you, with a big round of applause, our Pro-Chancellor Sir K.S. Jagannath Rao sir, for having such wonderful collaborations with all over the world scientists, we can say. So here, the participants who have the want to have the collaboration with Hegde, Professor Hegde, can contact Jagannath Rao sir, because they usually have the regular contacts, because I know personally sir, that's why I can say that he has a number of collaborations. So you can contact sir, and you can take the contact numbers also, and hope this session is useful to you, all of you. And thank you so much for spending your time with us till now, this session. Thank you so much.
See, very important aspect, as we said, if you listen, for many of you, is a very tough subject because you're trying to understand whole biochemistry. The important aspect is in brain, or in any body tissue, DNA repair is a very complex. It's not done by one enzyme, it is multiple enzymes. Our expectation like 1,000 enzymes involved in DNA repair. But still we have not discovered. Still we have not discovered. That's why we have no solution for cancer. We have no solution for the degenerating cell like Alzheimer's, Parkinson, whatever you say. So this is very important to understand. Major discovery of Dr. Murali, with the student, and we have two giant students when I was in Panama, that any repair enzyme is not independent. It has another friend to cooperate with that. So that cooperative system was tremendously worked. Actually, this is not done at all. This was the first study that has been shown. Now we know why in ALS, once you're discovered with ALS, you die in 4 years, 100%. When you detect with a breast cancer or ovarian cancer or prostate cancer, we know that cure is very low because we always detect them in a later phase. So we know that we give chemotherapy, we give radiation therapy, we give everything, but invariably we also know that we are losing the person in 6 months because the biology and the biochemical pathways in cell are so strong, so strong. Still we have to make discoveries. That's why in the poster session, we've opened a challenge for all of you. Come up with something new. Even you don't know about biology, you have not studied that pathway. Give a pathway. Give a pathway. Maybe it will be a genius pathway, we don't know. And the reason we would like to tell you, because today when we are having an intensive discussion after lunch with each group, we want to see maybe some of you come up with a new models. Maybe we'll be surprised. Such people will definitely encourage to publish your ideas as the best hypothesis in a top journals. Will help you, and maybe some of you want to continue that and want to do things, then we will find an opportunity for you also to work. Science works like that. Because when you want to collaborate, other partner always looks at you as a genius. You have something different ideas. If you simply say, I want to come and work with you, please give me an opportunity, nobody will give. Because they want to know what inherent potential you have that helps you to come. That's what in KL we are trying to build, not only in KL, outside KL also. That's why we'll be conducting meetings. We'll be spending a lot of our time, lot of our energy. To make this program, we have to spend a lot time. It's not money alone, but that makes us happy and satisfaction when we get at least from this group 5 best thinkers. We can help them to build our future. That's the main dream the institution is always focusing. That's what we need in the country right now. It is not just you pass an exam, you get some grade, you go away, and you get some PhD, do something, that's it's a different. But you want to be a discoverer, you need to have that. And I will close with one statement. After Sir C.V. Raman, we don't have a Nobel Prize winner. Because Sir C.V. Raman was genius. He's an observative scientist. Many of you don't know, when he was the director of Tata Institute, at 12 o'clock you see him in the roaming around the institute because he likes to watch the sky and see how the reflection of that light is working on that. So we need to have that kind of mindset people to have India Nobel Prize. People, Indians going outside, they won the Nobel Prize. But we need to have that. That comes from the younger minds. We have a lot of opportunity now in India to go outside, to build your ideas. You have MSME programs to build your knowledge, maybe your thoughts. And we have an international meeting coming up in Australia. Somebody has a beautiful mechanism. If you can able to explain, next year we'll sponsor some of you to young scientist meetings. But you have to be leader because the world is looking for leaders, not the students. Plenty of students in the world, no? What you will do with them? So what we are looking is the leaders. So today afternoon, I want to see that leadership with you people. Because if I identify at least two groups, extraordinary talented thoughts, I'll be the happiest man. The success symposium is over with that. I don't worry, other things. Success comes only when I see that thoughts. That's the future of science. That's the future of the country also. That's what we are looking for. Okay. Murli is one such example. When he was a student, he used to come, he used to sit with us till 10:30. He will have a dinner with us, and all the time he will be speaking only science. That's why today you see his papers in a very high impact factor journals, and he became a director when he was only 39 years, and he has got 3 RO1 grants. That means something like $8 million grant. Lot of students he spends. Even today he will work till 12 o'clock. That is his enjoyment. But he want to solve one problem. Why we cannot save an ALS patient or a Parkinson disease patient and make him to live longer. Okay. With this, I really waiting for the interaction in the afternoon. We want to spend lot of time with you people. Get you. There's nothing we are asking questions. There's no exam, nothing. What we want to thrill with your ideas. Okay. So we'll have a tea break and then we'll have a Japanese collaborator, Dr. Samuel Abraham from Japan, Emashin University. He's going to be speaker. He's going to speak on gut microbiome related to Parkinson disease, autisms. You see, fantastic new ideas is coming up. Thank you.
చేశాను సార్ అన్మ్యూట్ చేశాను సార్ ఆల్రెడీ ఇప్పుడు ఆన్ చేసుకోవాలి అన్మ్యూట్.
Can you hear me?
We are going to have the session very soon, within few minutes. And I request all the participants to move forward, and no participants are allowed to sit in the back rows, kindly.
Can you see my screen? I don't hear you. Can you see my screen?
Still more participants
Okay,
are there.
shall I start the talk?
Still more participants are there who are at the back. Kindly move forward.
Okay, so very good morning, everybody. Thanking Professor Rao, Dr. Hegde, and the colleagues and management of this institution and the organizers of the meeting for having given me an opportunity to share some of our work, thoughts, and opinions, as well as some suggestions for futuristic research in gut microbiome and brain disorders. So today, let me start giving you an account of 3 backgrounds and 1 tool that we were equipped with so that we can move forward. The first background, what I'm going to share, is about maternal gut health and neurodevelopment of the fetus. This is something that inspired us to do further research. These are all the data from the literature. And the second thing is alpha synuclein producing cells. And these were a great tool of learning, and that has given us an edge to further undertake research to move to a clinical side on this. And this was developed by Professor Hegde and Professor Rao. And the third thing is some of the lessons that we learned from evolution, that is gut microbiome connecting the animal kingdom and the plant kingdom. Among these 3 backgrounds, I will be going into details further. And the tool that we were equipped with is a polyextremo tolerant fungi, which is a very unique organism, and it is producing polysaccharides. So 3 backgrounds, I will first explain them to you one by one. The first inspiration or the background is about maternal health and neurodevelopment of the fetus. So this is one paper which very clearly says, early unsustainable and stunted development of gut microbiome is something associated, which is found in children with autism spectrum disorder. And another paper which reports when the maternal interleukin 17 in the mother mice was present, all the children, the offsprings born to that particular mice, they expressed autism spectrum like phenotype after they were born. There are several other papers, but 3 papers of similar nature were compiled together, and there was a perspective published in Nature in February 2021. And I'm going to explain each of these 3. The first one is here, where Curly protein and E. coli, when they are secreted in the gut microbiome, among the gut. This E. coli and E. coli induced protein, they move to the brain of an adult through the vagus nerve. They go and settle in the brain and they cause misfolding of alpha-synuclein in the brain and they cause Parkinson's disease. The second paper mentioned in this perspective was a filamentous bacteria which produces cytokines such as interleukin 17. If you see down here, the interleukin 17, they were able to cross the placenta of the offspring, and then they could cross into the developing fetus. They could even cross the blood-brain barrier of the growing offspring, the fetus. Then, when this offspring was born, postnatally, it showed symptoms like autism spectrum disorder. And the third one is related to motor neuron disease, wherein mucinephila accumancia and nicotinamide or vitamin B3, when it is secreted in the gut because of gut dysbiosis, they travel to the brain through the bloodstream, go into the brain, and then they cause motor neuron disease. So, both in adults as well as in a pregnant mother of a mice, they were able to predict or report the correlation between a bad gut or a gut dysbiosis where bad influence of not so good behaving gut microbiome could cause two diseases in the adult. One is Parkinson's disease and another is motor neuron disease. And when similar thing happens in terms of filamentous bacteria and cytokine production of IL-17, that can cause in the offspring of a pregnant mother autism phenotype. With these 3 points and neurodevelopment related diseases, I would like to, before moving to the next slide, I would like to point to the terminology called misfolding. Please remember that. Now, the second background is alpha-synuclein producing cell lines. So this is a publication by Dr. Rao and Dr. Hegde, and they were able to produce or induce in a specific cell line by genomic editing that they made those cell lines produce enormous quantities of misfolded alpha-synuclein in the lab. And this misfolded alpha-synuclein producing cell lines, they were able to help implement or develop or produce Parkinson's disease models so that you can try different therapies. So alpha-synuclein is a neurotransmitter, and misfolded alpha-synuclein producing cell lines could be developed, and these cell lines were used in mimicking Parkinson's disease in animal models. Now the third background is what we learned from the evolution. This is the tree of life in a very simplified manner. Importantly, the animal kingdom and plant kingdom have been depicted, and the fungi are placed in between, interposition between animal and plant kingdom. And I would say this fungi are connecting animal kingdom and plant kingdom through our gut microbiome, because animals and plants, they have gut microbiome or microbiome in their body in some form or other, and these are connected. And now if you look at the fungi, going by the history of medical inventions, one of the first very successful antimicrobial, there is a penicillin, is derived from a fungi. One of the earlier anti-cholesterol lowering agent, a statin, was from fungi. And also several anti-cancer agents have been developed or produced from fungi. And based on this, animals and plants who live on surface of earth are having gut microbiome, and the fungi are connecting us through our environment to the environment using this gut microbiome. Now, the tool that we were able was two different variants of polysaccharides or beta glucans produced as an exopolysaccharide by a black yeast called Arabasidium pullulans. And this was initially looked upon by a team of zoologists and agriculturists. Now, on the left side, what you see is a picture. The central glistening or a core center particle is a living organism called Arabasidium pullulans, and surrounding this central glistening area, what you see as a halo are the exopolysaccharide kind of produced of this beta glucan, and this is a produce of this yeast. So this exopolysaccharide, when we evaluated, there are two strains we have evaluated. So both, in spite of having the same chemical formula, structurally have a different structural identity. So this is a tool that we were gifted by nature, and with this we started a research, and these polysaccharides are produced in Japan in a place called Kochi in a GMP facility in bioreactors like a drug like this. Now, we started a clinical study in children with autism spectrum disorder. Why? Because a clinician, developmental pediatrician, after going through these left side, this I had already explained to you, the three different diseases caused in human beings by gut microbiome dysbiosis. One of them even crosses the placenta of the child of a child of a pregnant mice, and this, when it causes autism spectrum disorder, this autism spectrum disorder children clinically are having constipation. On the right side, this yeast produced exopolysaccharide as a food supplement. 22% of the people, they were consuming this for relief of constipation. So, Dr. Raghavan, the clinical investigator of the study in autism spectrum disorder, he chose children with autism spectrum disorder who have constipation to be included in the study. And the study, when we conducted, originally we thought we should evaluate only constipation. Initially, 18 children were chosen. They were screened, they were randomized 1:2, one portion, one candidate for control, and two candidates for the treatment arm. The control treatment comprising of remedial behavioral therapies and L-carnosine, there is a standard drug they were all consuming, 500 milligram per day. On the right side, along with the conventional treatment, this particular beta glucan, they consume 0.5 grams twice per day. It's in granule form. They add it to yogurt and then consume. Now, the first feedback we got from their mothers or one of the parents was that these children, they started sleeping well compared to before the study, and even the sleep pattern was much better even in the compared to the control group. It was better in the treatment arm. And then we measured the serum melatonin. The serum melatonin increase in the treatment arm was much better compared to the control group. And melatonin as a food supplement or as a drug is prescribed in several countries. However, in these children, without any external supplementation of a melatonin, either as a drug or as a food supplement, their endogenous melatonin quantity increased. This is something very phenomenal. And this paper has been published in the journal Brain and Behavior. And then we wanted to evaluate the alpha-synuclein, the plasma alpha-synuclein in this children. Why? Because there have been varying statements or reports on levels of alpha-synuclein in children with autism spectrum disorder. This is one paper by Al-Mazidi from Egypt. They say even the plasma levels of alpha and gamma synuclein could be considered as an indicator of severity of the autism spectrum disorder. There is another paper where they say they have reported detecting alpha synuclein aggregates in the gastrointestinal biopsy and by protein misfolding and cyclic amplification in these patients. Now, interventionally, invasively, if you want to do a gastric biopsy, it is quite tough. It is not permissible because the risk is high. So we chose only to evaluate the plasma levels of alpha synuclein in these children. So if you see on the right side, the baseline to the 90-day study after the 90 days, the control group did not have much difference, whereas in children who consumed this polysaccharide beta-glucans, exopolysaccharide beta-glucans, it increased 2.5 to 3-fold. And also the CAR score, that is the childhood autism rating score, which is based on the behavioral pattern change of these children, between these two groups, it improved. Lesser is the score, better is the improvement in terms of behavior. So you see here in the beta-glucan group, polysaccharide group, the score came down. That means the severity came down. So it was an indicator of an improvement in terms of behavior of these children. So this is published in BMJ Neurology Open. Now the most important point is we evaluated by RNA metagenome sequencing the control group and also the treatment arm comparison of their gut microbiome. Most importantly, the Enterobacter and E. coli. You see the Enterobacter compared to the group 1 on the left to the right, pre and post, after the intervention, they almost became zero, almost became nil. The E. coli also, compared to the control group, they decreased significantly. There have been interventions to value add or contribute to the gut microbiome eubiosis in terms of probiotics, wherein either killed organisms or live organisms you consume. There is also another treatment called fecal microbiome transplant, wherein healthy donors' fecal microbiome are colonoscopically transplanted to people who have certain diseases. On those cases, either a good bacteria is added in terms of a probiotic or a healthy gut microbiome composition as such as a cluster is transplanted to somebody with a not so healthy gut or with disease. On both cases, you are adding something good to somebody who is ill. Here, we have added something which is bringing down the bad gut microbiome. This is something very phenomenal, we believe. So, this exopolysaccharide secreted beta-glucans, they somehow have the manipulating capacity not only to improve the good quality gut microbiome, but also they were able to control the bad ones in the gut. And this paper was published in the Journal of Alzheimer's Disease, wherein we wrote a hypothesis, and when we look at the genome sequencing result, the abundance of harmful enterobacteria decreased almost to zero, and harmful bacteroids also decreased in the beta-glucan exopolysaccharide consumed group. The enterobacteria secretion of Carli that causes misfolding of amyloids, such as alpha-synuclein, whose accumulation leads to inflammation and neuronal dysfunction that is central to pathogenesis of autism, Parkinson's disease, and Alzheimer's disease, and decrease was seen in harmful bacteria like E. coli, Akkermansia muciniphila, CAG 154, Blardia species, Coprobacillus, and Clostridium bolte, with an increase of beneficial bacteria like Faecalibacterium, Brasiliensis, and Privitella copri. Now, in this image, what we have uh, reported is the enterobacteria which secrete Calais amyloids, they are causing alpha-synuclein misfolding and their accumulation in the enteric neuronal system. So that's what in one of the earlier paper I had showed, the gut deposits of this misfolded alpha-synuclein has already been reported. So the enterobacteria, when they are more, they secrete Calais and amyloids. They make the alpha-synuclein into an abnormal misfolded alpha-synuclein, and this misfolded alpha-synuclein aggregates in the gut, cause dysbiosis, and then they move to the brain through this gut-brain axis, vagus nerve or a bloodstream, and then their deposition in the brain can cause Parkinson's disease, dementia, and Alzheimer's disease. It is also a known fact that children with autism spectrum disorder, they are more prone to develop Alzheimer's disease and Parkinson's disease in their late part of their life. Now, we hypothesize 4 different steps wherein these exopolysaccharide beta-glucans could work, or they likely to work. They control intrabacterial growth, therefore block Calais secretion, alpha-synuclein misfolding and its deposition, which is the root cause of the disease. The second thing is, it is already known that these beta-glucans in healthy human volunteers and disease patients, they have been found to be reported to have increase their natural killer cells, endogenous natural killer cells. So natural killer cells also have the capability to scavenge this Calais and amyloid deposits. Number 3 is they balance the gut microbiome, they control dysbiosis and increase the beneficial gut microbiome as a prebiotic. And also, there have been reports wherein this exopolysaccharide produce beta-glucans. They, through the GABA mechanism, they can rejuvenate the microglia, help clear the deposits in the brain. There is only a hypothesis. With this, we have not done a study, but in future, we are planning to do some animal studies on microglial activation of these beta-glucans in animal models. Now, further research also is necessary on this, because since some people have reported increase in alpha-synuclein in the plasma of children with autism, some have reported decrease in alpha-synuclein. So instead of the quantity, what we foresee or what we forecast that we should be done is, we should evaluate the quality of the alpha-synuclein, whether the alpha-synuclein which is circulating in the plasma of these children with autism spectrum disorder are either normal or abnormal, such as misfolded. For this, I think we should take some inputs from Professor Rao and Dr. Hegde. Now, coming to misfolded proteins. So our research further moved in this direction of looking into where all misfolded protein related diseases are happening, or which are all the misfolded protein related diseases where these exopolysaccharide secreted beta glucans may have a role to play. Now, before I go into the next topic, I'll just give a brief explanation on this misfolded protein. This is a cell. Each cell has a task of producing some proteins in general. For example, if it is a beta Langerhans cell, they are supposed to produce the insulin. Thyroid cells should produce the thyroid hormone. So each cell has to produce some kind of protein. Now, if you see a cell has a nucleus, they produce the protein, and this protein is folded for a proper delivery as an exocytosis. And for the delivery, the endoplasmic reticulum, it's a kind of an assembly. It's a manufacturing system. So the endoplasmic reticulum makes the proteins folded in a proper shape or size or in a manner in which they can be carried as an exocytosis outside for delivery into the intracellular fluid, so it can be carried by the bloodstream to be delivered to some other organ. Now, when this endoplasmic reticulum, due to some disease or some genetic problem or some stress because of lack of nutrition or something, or because of infection, if the endoplasmic reticulum undergoes certain stress, the folding of proteins doesn't happen properly. So if it is not happening, then the cell employs a system called unfolding protein response and then tries to settle it down. So if it is not going to happen, then the misfolded proteins kept on getting secreted. Misfolded protein is an abnormal protein, so they are either going to be useful to the organ, or they can easily be carried away the bloodstream. So in case of alpha-synuclein in the gut, because of the external influence by the gut microbiome, bad gut microbiome, producing Curlian amyloid, alpha-synuclein becomes misfolded. Instead of folded normal alpha-synuclein, it gets misfolded. And this misfolded alpha-synuclein not carried by the bloodstream, so it gets deposited there. So now, with this exopolysaccharide beta-glucans, we were able to do some manipulation of the gut microbiome. Exact mechanism, though we don't know. Probably the gut microbiome control is preventing such misfolded alpha-synuclein production, and therefore their unwanted aggregation. So it goes well. So we looked into where all this kind of misfolded proteins are happening. So one such misfolding of protein, it's my area of cardiology. They are very clearly documented. Protein misfolding leads to certain cardiac diseases. And also in terms of type 2 diabetes, proinsulin misfolding is one of the early events that leads to diabetes. So these are all very broad areas where there are already remedies available. Heart diseases, cholesterol lowering agents, anti-diabetic agents, all this are available. And we were looking forward to a target where there is no therapy or no definitive solution available. And that is where we came across missense mutation in dystrophin in Duchenne muscular dystrophy. If you can read here in this paper, they have evaluated 4 mutants that cause dystrophin mutation, and dystrophin mutation, they found an association to amyloid diseases, and they also say that this dystrophin mutation made serious misfolding and aggregation. So misfolding of dystrophin protein due to missense mutation has been conceived as a role playing agent in terms of Duchenne muscular dystrophy. Now let me tell you something about the Duchenne muscular dystrophy as a disease pathology. In Duchenne muscular dystrophy, in the muscles, dystrophin is a protein which is present in the muscles that makes the smooth functioning of the actin and myosin filaments of the muscles. It's like you can say in a broad terminology, in a layman's terminology, I will say dystrophin is a kind of a lubricant, let us say. Now this dystrophin missense mutation, or genetically the dystrophin producing protein is defective, so dystrophin is not produced. Because dystrophin is not produced, the movement of the muscle fibers becomes very rough. Because of the rough movement, there is an inflammation, and because of inflammation there is a damage causes the muscle cells to degenerate, and degenerating muscle fibers need to be replaced. So the satellite stem cells in the muscles, they come in aiding, they proliferate and then try to replace the cells which are lost. I mean the muscle fibers which are lost. Then this inflammatory surroundings is not allowing those new satellite stem cells to become muscle fibers. Instead, they all turn, because of the hostile environment of inflammation, they become fibroblasts, and then they settle as fibrin, and then they become it leads to fibrosis of the muscle. Due to fibrosis, the muscle movement becomes very rough, and dysfunction causes these children to become non-ambulatory. Their locomotion is affected, and over a period, the respiratory muscles are affected, cardiac muscles are affected. And what happens in actual life is, most of the Duchenne muscular disease children, they start exhibiting a difficulty in moving their limbs between the age of 5 to 7, and they are unable to get up or walk. And genetic mutation, once it is diagnosed, then they start on certain drugs which are only be able to manage the symptoms. Not definitive recovery is possible at this point of time, because only recently a genetic disease modifying gene therapy, not disease modifying gene therapy, it's a definitive gene therapy has been approved, but we have to wait and see how effective it is. So in the early teens, most of these children between the age of 12 to 15, they become wheelchair bound because they cannot walk by themselves. And then in their mid-teens, around 15 to 20s, they develop respiratory problem because the respiratory muscles become weak. Then in the 20s to late 20s, they become bedridden, or they become victims of cardiac disease because the cardiac muscles are also getting affected. And most of these children, they don't live beyond the age of 30. In the 1970s, the average lifespan of these children born with Duchenne muscular dystrophy was between 18 to 20 years, and after supportive and symptomatic disease modifying therapies, the lifespan extended to 25 to 32. Today it's between 28 to 32 average. Now, if you see the therapies that are available, gene therapy is a definitive therapy which has only recently been approved, and it costs approximately $3.2 million. Exon skipping therapies were in place for at least 5 years ago, and now exon skipping therapy again costs between $500,000 to $1 million, say 2 to 5 crores a year. Disease modifying therapies are the mainstream of the therapy, in which we use, most of them use certain antibodies and steroids. Steroids control inflammation. However, due to certain adverse reactions, steroids cannot be continuously administered to the children. Sometimes we'll stop for a while and then restart, so it happens like that. Cell-based therapies are in clinical trial, where new stem cells or iPS derived cells, they are trying to inject into the muscle so that the damaged or degenerative muscle cells can be replaced or rejuvenated. It is still in clinical trial. Angiogenesis therapy is still evolving. They are trying to use cells or angiogenetic agents so that more blood flow will go in, clear the fibrosis, clear the macrophages, clear the fibrotic causing fibroblasts, give new blood flow, new stem cells, new muscle cells, but that is also in the clinical trial. Now the key words are inflammation and fibrosis. So coming back from what we started, misfolded proteins are causing or contributing to Duchenne muscular dystrophy. Because of that, inflammation and fibrosis is happening. So we thought, why not we try this exopolysaccharide beta glucan? We had different strains in our lab, in the library. So why not we develop or choose or try if there is something that we have which can control inflammation and fibrosis? That is where we started our research. So we screened our library, and from there we took one particular organism, that is Arabidopsis fully the same organism, one particular novel strain of this organism, which was able to produce a structural beta glucan 1,3, beta 1,3,1,6 glucan as a polysaccharide in the laboratory, and also we could scale up to the industrial scale. When we compared it with the previous strain, this as an inflammatory marker, non-esterified fatty acids, this was able to control very well in the KKAY mice, which is a diabetic animal model. Thereafter, we evaluated what is called leukocyte to CRP ratio, LECR. And this LECR should increase in terms of immune modulation. So that was a good improvement. So this particular strain production was able to give rise to an improvised LECR in Sprague Dawley mice. Then we did a study comparing these two in middle-aged healthy Japanese volunteers. There we found an improvement of LECR and also all the inflammatory modulation, especially control of D-dimer. Then we did a study in patients with COVID. In 30 days, we were able to prove that this particular strain, when produced, exopolysaccharide produced beta-glucan, when it was consumed orally, it brought down interleukin 6 and also D-dimer levels. This was in 30 days. This has been published in Biomedicine and Pharmacotherapy. Then we did another very insightful animal study of comparing these two beta-glucan produced separately and together. What we found was this AFO2O2 produced was a good prehepatic metabolic balancing agent. It controlled abnormal glucose, it controlled abnormal lipid levels, and then this N163 produced, it was able to, as an independent agent, control the inflammatory response in this NASH animal model. NASH stands for non-alcoholic steatohepatitis. This is a very inflammatory model where these animals over a period, you keep feeding high fat diet, they will develop fatty liver, then move into fibrotic liver, then cirrhotic liver, and then they will develop hepatocellular carcinoma. In these hostile environments, this N163 produced beta-glucans, they were able to control the interleukin 6 at the plasma level, and they were also able to reduce the liver fibrosis and liver inflammation proven by sections of this tissue, and which was published in Journal of Experimental and Clinical Hepatology. Then we evaluated the gut microbiome and fecal metabolome. Interestingly, there was a very good increase, that is, the gut microbial diversity increased greatly in the when we administered these two together, and post intervention the abundance of firmicutes decreased whereas bacteroids increased, and was the highest in the combination group. You can see this data here. And then if you go individually, the enterobacteria decreased in A4202 and it decreased in A4202, Turicibacter in N163, Bilophila in N163, and also Prevotella, Lactobacillus, Proteobacteria, Akkermansia, they all had a good outcome in terms of a beneficial reconstitution. Then, if you look at the gut metabolome regulation, succinic acid, phosphoric acid, fructose, isoleucine, leucine, phenylalanine, methionine, spermidine, ornithine, they all gave a very good indicator that either A4202 alone or N163 beneficially was able to modify these metabolomes in the fecal material which from these animals. Then these were all correlating to one thing, that is inflammation and fibrosis we are able to control, evident by not only the gut microbiome but also the metabolomes. Then we got the study and approval by the ethics committee for doing a short duration 45-day duration study. It was a two-group study. And again, the Duchenne muscular disease patients were randomized 1:2, control arm 9 and treatment arm 18. Importantly, this being a food supplement, we didn't want them to stop any of the drug because this is not a kind of replacement to any drug. So we made them consume whatever the drug they were consuming prescribed by their neurologist or physician or pediatrician. Along with that, they consumed this particular exopolysaccharide beta-glucan in the treatment group. The control group, they had routine physiotherapy, calcium, vitamin D with or without steroid, because the steroid, some of them consumed, some of them did not, some of them were intermittently consuming as well. On the treatment arm group, they were all the determined medications, routine medications as a standard of care they were consuming. Along with that, orally they consumed this exopolysaccharide produced beta glucan. So we looked for plasma markers interleukin 6, interleukin 13, TGF beta, and plasma dystrophin. We evaluated the muscle function by medical research grading of muscle strength, and those who were able to walk, we subjected them to 6-minute walking test at baseline and after 45 days. And importantly, we evaluated their gut microbiome at the base level and also after the 45th day and subjected them to metagenome sequencing. If you see the plasma inflammatory markers, there is very good control of interleukin 6, interleukin 13, and also TGF beta. And the MRC, there is a very slight, not so significant improvement, but for 45 days, this is something very good. But there was no significant change in the 6-minute walk test or the North Star ambulatory assessment because it is a very short duration. Then, before moving to a long-term clinical study, we did an animal model study in MDX mice. Inflammation and fibrosis were evaluated by muscle biopsy and histopathology. Muscle regeneration were evaluated by CD44 and MOH3 because in these children, 45 days or 180 days, subjecting them to a general anesthesia, a surgical procedure of intervening invasive muscle biopsy, it is quite risky or it's quite tough for them to undergo. So instead of human studies, we did in preclinical animal model studies, we took the biopsy of the skeletal muscle of these animals. And you can see here the normal one, vehicle one. Compared to that, this exopolysaccharide beta glucan ones had a very low inflammatory tissue pathology, and also fibrosis also was much lower in these animals which consumed this. So we have a hardcore proof of decrease in inflammation and fibrosis. So when decrease in inflammation and fibrosis, we have to look for increase in muscle regeneration going by the earlier data I had presented. So it is the inflammation and muscle fibrosis, they make the newly regenerating stem cells not to become muscles, but instead they become fibroblasts. But here, because of the reduction in inflammation and fibrosis, the increase you can see on the rightmost part, both by CD44 marker and myosin heavy chain 3, these are all markers of regenerating muscle cells. So you can see on the right side, whichever the group that consumed this exopolysaccharide beta glucans, the muscle regeneration was higher. Then we undertook a 180 days study in which we did only one group, 12 ambulatory and 12 non-ambulatory. They all, along with the conventional treatment, consumed this, and the NSAA, MRC, and 6-minute walking test, there is a slight improvement. Usually in these children, even year after year, month after month, the baseline to walking distance will keep on coming down. But in our case, the 6-minute walking test average, though there is no statistical significance, it didn't decrease. Not only that, average was increasing. It is a very good sign. This is a path-breaking outcome. Now, in the 45 days clinical study, when we evaluated the plasma dystrophin, that is the dystrophin produced by the vascular smooth muscle, it increased by 32%. What does it say is, the disease is a genetic disease. Lack of dystrophin in muscles is what it manifests. Gene therapy, exon skipping are still very expensive. Some of them are in trials, and the clinical efficacy is not more than 5%, whereas dysfunction of the skeletal muscle, respiratory, cardiac muscle as a manifestation is controlled by disease modifying agents. In our case, without any adverse reaction, we were able to control fibrosis, control inflammation. Increase in plasma dystrophin means the vascular smooth muscle produced dystrophin also have increased. Not only the skeletal muscle produced dystrophin. Therefore, the more the blood flow is possible to improve the muscle regeneration and control the fibrosis. So this exopolysaccharide beta glucans are able to yield control of inflammation, control of fibrosis, improved regeneration. And with this, we are hopeful that the lifespan of the children could improve. And importantly, when we looked into their gut microbiome, that gut microbiome also had phenomenally improved in these children. You can see here Enterobacteriaceae, Firmicutes, Akkermansia mucinifila, and Alice types it decreased. The bad ones had decreased, but the good ones like the Lactobacillus, Bifidobacterium, Pruvitella, and Rosperia had increased. This is a good sign. That is why we say, if you look at this graph on the left side, children born with Duchenne muscular dystrophy before the year 1970, they had a lifespan of 18 years, average lifespan. Between 1970 to 1990, it was between 22 to 24. Post 1990, it increased to 28 to 32. By this disease modifying, this is a speculative curve I have drawn. Probably we'll be able to increase their lifespan by at least 10 to 15 years. Because their gut microbiome changed, probably, I do hope, has played a key role in bringing the anti-inflammatory and anti-fibrotic effects in their skeletal muscle and also vascular smooth muscle and cardiac muscle. Cardiac muscle we are evaluating now. Then we move to another neurological disease, which is a very tricky and complex disease called multiple sclerosis. We did a 60-day study. Why we took multiple sclerosis is because multiple sclerosis is a complex disease where the exact pathology, causative pathology is not known, and an unknown hyperactive immune system has been acting against an unknown enemy. So this activated hyperactive immune system is attacking the myelin sheath of the nerve, different nerves. Some patients with multiple sclerosis, they may have a generalized functional problem. Some may have only their eye nerves of the eye affected. Some may have their throat nerves affected. So each patient will have a different kind of manifestation because the common problem is there is a hyperactive immune system attacking the sheath of the nerves. Nerves are damaged. And in these patients, we did in 12 patients between 24 to 68 years of age, conventional treatment. Along with that, they consumed 2 sachet of this orally consumable exopolysaccharide produced beta glucan. We evaluated inflammatory immunological biomarkers, expanded disability status scale, which is a gold standard of evaluation in these patients, and importantly, the gut microbiome. Why gut microbiome in these patients also? Because this is one neurological disease where, along with the increase in severity, the gut microbiome correlation has been clinically documented. They have documented that the bad gut microbiome increase in these patients is correlating with the increased severity of the disease in multiple sclerosis, because again, this disease, sometimes it can be progressive, sometimes it can be keeping on increasing severity and then go into remission and it may relapse. So it will be like up and down for this patient. So they have evaluated the gut microbiome of these patients, human patients, at different stages, and they have proven that improved quality of life is correlating to decrease in the bad gut microbiome. In these patients, when you look at the data, relative abundance at the phylum and genus level, actinobacteria and bacteroidetes, pre and post, you can see the post intervention actinobacteria significantly increased, bacteroides have decreased. Same way, the genus level, pre and compared post intervention, you can see the bifidobacterium have significantly improved and also certain prevotella. At the same time, relative abundance also increased. Then at a species level, you can see compared to the pre, on the top is the post intervention, Prevotella caprai significantly increased and Faecalibacterium also increased. If you take a specific species level, Bifidobacterium longum, it increased. Prevotella caprai also increased because this Prevotella caprai, many studies has been reporting that either reduced abundance of Prevotella in relapsing remitting multiple sclerosis or an increased abundance after treatment with disease modifying therapy. So other disease modifying therapies also yielded an increase, and this intervention also has improvised this outcome. That means this is a good system that we have employed. Same way, Faecalibacterium and Syphilis also post-intervention increase, which is also a correlating factor to the betterment of quality of life in these patients. Whereas the bad ones like Blottia, Doria, which are causing the disease condition to worsen, they came down, and the interleukin 6 from 27.4 came down to 9.67, which is an important cytokine which is correlating to cytokine storm in COVID patients or increased inflammatory evidence in patients with multiple sclerosis. Now, we undertook another study in Parkinson's disease patients, which is now again going back to my earlier data, misfolding alpha-synuclein accumulation in the brain. We did a study, but I'm not going to go into the details of the study because I think we're running short of time. One important paper from Finland says antibiotic exposure increases the risk of Parkinson's disease development in patients, because they did a study of evaluating retrospectively 13,000 to 15,000 patients with Parkinson's disease, age match control another 40,000. What they say is gut microbiome alterations have been found in prodromal and established Parkinson's disease. What the hypothesis is? Unwanted use of or unscrupulous or overdosage of antibiotics. Antibiotics is necessary, but when one is exposed to antibiotics, it's more than what he or she is supposed to be. That causes a gut microbiome alteration, leading to probably Parkinson's disease. And now, finally, we wrote a hypothesis. Gut microbiome are going to help in the regenerative medicine also. When you want to regenerate the neuronal system, several of the diseases are caused by accumulation of unwanted proteins like alpha-synuclein or Levy bodies or amyloid bodies. Now, they have to be cleared. The scavenging of amyloid deposits through microglia is something. Here, there are three components wherein gut microbiome provides indirect or direct support to all three modes of regenerative medicine mechanism. One is direct replacement of cells. They recruit progenitors, stem cells, and immune cells indirectly, or they can provide paracrine support. See, the short chain fatty acids, nitric oxide, neurotransmitters, lipopolysaccharides, metabolites production, especially the butyrate production by these exopolite beta glucans, can improvise gut microbiome environment to indirectly help the regeneration of neurons. And as a scaffold, indirectly they can help beneficial modulation of extracellular matrix by regulating inflammation and secreting of factors of relevance which act as scaffolds. Finally, I would like to close with this slide, wherein this is a hypothesis which is published recently by our group. On the left-sided image, within this image, I've got two components. The left-sided blue background is an environment of the earth where the crust of fertile land is hosting a plant, and the plant is bearing fruits, which I have compared onto the right side to the gut microbial environment. You see the root of the plant within a few meters. It is supported by the crust of the fertile land. The land has to provide essential nutrients, including water, but it should be devoid of toxic substances. And also, you should have enough sunlight, atmospheric air, rain, water, which is provided by the atmosphere, so that the plant is growing to the necessity vegetative growth and starts yielding fruits. Similarly, on the right side, I am comparing as an analog the crust of the fertile land to the intestinal epithelial cells. Intestinal epithelial cell should be nourished with blood supply of good quality nutrients, enough of fat, enough of carbohydrate, enough of protein. They should not have more sugar, more bad lipoprotein, or whatever it is. Same way, toxic elements should not be there. Now, when the intestinal epithelial integrity is maintained in a proper way, good microbiome will thrive. And this good gut microbiome not only are supported by the intestinal epithelial cells, but they also derive the flow of food material, digested food material from the gut, from the stomach to the intestine and then towards the rectum. They are all nourishing this gut microbiome. So nutrition from the digested food and intestinal epithelial giving a supporting environment and nutrition, well optimized good quality nutrition, good microbiome thrive and produce good metabolites, short chain fatty acids. So there are two kinds of exopolysaccharide produced beta glucan I was telling, or polysaccharides I was telling. One is the A4202, which is a good metabolic regulator that ensures a good quality nutrition both within the gut and to the intestinal epithelium, maintaining its integrity and its function status, whatever structure. Whereas the N163 beta glucan is a good immune modulator. It doesn't allow overactive cytokine storm or overactive immune system, or it's like a weed killer, if you say in a layman's term. Maintains a balance so that it maintains the balance within the gut as well as from underneath the gut in terms of epithelial integrity. This provides this. So we need to do more research on the exact mechanisms. To summarize, I would say homeostasis of gut microbiome, that is maintenance of the eubiosis of the gut microbiome, is the most important criteria towards endurance of a healthy long life. So we need to do more research on two important points. One is how a good quality gut microbiome are playing the epigenetics of several diseases, especially the genetic diseases. The second thing is, as I had mentioned, overdose of antibiotics is killing the gut microbiota, the good or they cause dysbiosis. So there should be some focus on evaluating the toxicity of any food or any drug to be based on an additional criteria of their implications to gut microbiome eubiosis. So if you do that, if you check whether it is a good food or a bad food, good drug or a bad drug, taking into account their adverse implications on the gut microbiome, then they are very, very, very safe. Thank you very much for your time and opportunity. So now I think I finished within 55 minutes or something. So I'm open to take up questions, whatever you may have. Thank you. I am not audible. Am I audible to you? But I'm not. I don't hear you. Can you speak? Can you just test? Hello, Professor Rao.
Yes, Chandvasha, type చేయమ్మా.
Okay, I could see, I could read the questions in the chat. Yes, please let us. You can type chat, and I'll be. Yeah, please ask the question through type chat. Thank you. Please ask your questions through the type chat. I'll be answering.
There is a question which has come, sir.
Regarding the fecal microbiome transplantation that you mentioned, may I know your experience towards the potential? Well, fecal microbiome transplant, to my knowledge, what I know is there are two kinds happening. One is like a drug which is under evolving. They have not approved it yet, but it's in the clinical trials. But what I understand is there are several hospitals, even in the United States, they take the healthy donor fecal microbiome, they don't culture it in the lab. They just transplant it. That's what I know. And culturing, yes, quite it's difficult. So you can Google or you can PubMed fecal microbiome transplant. There are several publications. Usually, what they do is that they take it from the spouse or the family member who is healthy and then try to transplant it colonoscopically. That's what I know. I don't think they culture. Yeah, metagenomic sequencing will give a whole spectrum and abundance of different microbes, which may not be all culturable. Yes, I agree. Because there are hundreds of thousands of organisms, at least to our knowledge, we know at least 100 different varieties. Among them, the ones which are causing the influenza, only say hardly we know 10 or 20 different kinds. So if we can isolate them, culture them, that's good. But when you culture them, but the numbers may be good, how far their functional capabilities will be, that we will have to do. We'll have to study. Thank you. Hope I have answered your question. Any more questions? You can always type chat here. Yeah, next question is, can you share some light on whether fecal microbiome has shown any effect in overcoming, I mean, age-related macular retinal disease, or can you?
Yes, dear participants, you are welcome to ask questions.
Antimicrobial resistance. I don't know. Don't have any idea. Probably they can try some, they may have to do, but I haven't gone through any papers on this. But antimicrobial resistance, yeah, it's a major problem. I think there are some clinical reports, but I'm not sure whether it has been standardized or not.
The participants are requested, if at all you have any questions, kindly type over here so that your question will be reachable to the speaker and you will get clarified the questions with answers.
So when they find probably multi-drug resistant bacteria-related disease, probably a fecal microbiome transplant from a healthy donor would be something that is worth trying clinically and then see how effective it is.
Sir, you are muted, sir. We are not able to hear, sir.
Yeah. Apart from glucans and short-chain fatty acid, any proteins released from healthy gut microbiome? Well, glucans, I didn't say that they are released. We made them consume specific beta glucans, and then these beta glucans nurtured the environment, made good gut microbiome. Gut microbiome made a contribution in terms of good short chain fatty acids or proteins or metabolomes. Importantly, butyrate. Butyrate is one of the important components we found in our study, because butyric acid or butyrate, is considered to be a correlating factor of relevance to longevity. You can find in the literature, there was one study by Professor Naito. They compared the gut microbiome, metabolome, various blood-related parameters of a group of healthy, long-living centenarians in Japan. Because that area, the people live longer. Every third person is above 100 years. And then they took comparatively another region in Japan where relatively the lifespan was short. And they compared the parameters, all parameters of relevance, biological, biochemical, and everything. What they found as a single most important demarcating factor between the centenarians and the shorter lifespan group was the centenarians had very high level of butyric acid in their gut fecal metabolome. So these are all some of the key factors that indicate that we need to make our gut environment good. Then we need to make a good arrangement or an understanding of their gut microbiome, so that they help us live longer. Any more questions? Okay, on which factors good and bad gut microbiome decided? Well, if you allow me to point to one factor, that will be the epithelial integrity. See, the gut microbiomes, they all thrive because of the intestinal epithelium. And there is a paper or even a book which says leaky gut or inflammatory gut epithelium, the mucosal membrane epithelium, is the fundamental beginning starting point of any and every disease. So leaky gut, so inflamed gut. Inflamed gut means the intestinal epithelium should be kept healthy. So for intestinal epithelium to be kept healthy, as I had mentioned, you need to give them good nutrients. So you need to be very careful about what you eat. And again, your metabolism has to be under proper way of maintenance. Epithelium is very, very important. Epithelial integrity, if somebody can maintain well, then that makes the good microbiome survive and doesn't allow the bad microbiome to survive. May I seek your kind suggestions to present youth with respect to gut prevent consuming spicy food, junk food, and alcoholism? Yeah. Yes, that's why I already said good food is very important. So that there is a saying which I can translate in English. When you are young, if you eat very carefully your food like medicines, when you get old, you may not have to consume medicines like food. So we have to be very careful of what we eat. Yeah. So thank you very much for your time, this opportunity, and especially to um, Professor Rao, Dr. Hegde, and the management of the university, and especially for the C.V. Raman Award. And I think I do hope I am worth it, and I'll try to come up to your expectations and contribute to further research. So thank you very much, and I wish the event go well, and then would like to future collaborate with Dr. Hegde and Professor Rao in the future. Thank you very much. Bye bye.
That's a wonderful presentation by Professor Sam Abraham sir, and we are very much thankful for his wonderful presentation, and which is very valuable. And the question and answer session also was, as at present generation they are facing many problems with the malnutrition, as well as Subakar Raju sir mentioned quite uh, suitably, that spicy junk foods and how especially the food which we take affect our brain was quite well known with the presentation by Professor Abraham sir. Thank you very much sir. On this occasion, I'm very proud and honored to announce that the Dr. C.V. Raman Award was presented to Professor Sam Abraham sir by KL authorities, and distinguished best speaker award also. Thank you so much on behalf of KL management in his absence. Once again, with a round of applause, dear participants, with a round of applause, I request all of you to thank Professor Samuel Abraham sir for his valuable presentation. Thank you so much, sir. And ladies and gentlemen, dear participants, it is really very glad and very privileged to announce that amongst us, wonderful personalities today, and we are very fortunate to meet them today. They are none other than, I'm very much delighted to introduce as well as to invite Dr. Ramakrishna Garu, the Regional Director of NSS, and Dr. P. Ashok Reddy Garu, State NSS Officer, onto the dais. May I humbly request Dr. Ramakrishna Sir, Regional Director, NSS, and Dr. P. Ashok Reddy Sir, State NSS Officer. And now I humbly request our Pro Chancellor Sir, K.S. Jagannath Rao Sir, and NSS Director, Dr. Srikanth Reddy Sir, and Student Affairs Advisor, Subakar Raju Sir, to felicitate our guests. I humbly request Sripati Rai sir also to be on the dais to felicitate our chief guest today, Dr. Ramakrishna sir, as well as Dr. P. Ashok Reddy sir, with a token of memento and shawl. And I request our participants to give a big round of applause on this occasion. Sir, it's really indeed a great pleasure to us to meet you. We are very happy and glad to meet you and felicitate you on this occasion of 5-day workshop, international workshop. Thank you so much for being with us. Thank you so much, sir. As you know, the NSS National Service Scheme, a really wonderful job done by the NSS volunteers in our campus as well as all over India. And now we are very much thankful to the Regional Director, Dr. Ramakrishna sir, as well as Dr. P. Ashok Reddy sir, State NSS officers, for giving their utmost support to our authorities as well as KL students. Thank you so much, sir. On behalf of the management, I would like to thank both of you for being with us on this occasion. Thank you so much, sir.
So Samuel Abraham highlighted you about the gut microbiome. I know you're all hungry, and gut microbiome is a very important, as you know that two things. You should always account. First one is if you are stressed, if you are depressed, and you don't have a good mental health issues, your microbiome culture is going to change all over your body. You have to be very clear. So you have to maintain a good mental health parameters to keep your microbiome. We always say, you when you meet people, some people, some people very sweet. We call them sweet people because they smell different. We call fruit smells. The people with the positive mindset, happiness, they produce what is called amines in their sweating. Wherever they sweat, they produce what is called amines. That's why they always looks in a smell. Some people smell very odd because of the compounds they smell. It's called amides. So amines and amides makes a difference in your personality. And many of us, when we meet, many of the people we meet, we also smell their mouth. You cannot withstand longer time, because of the positive, negative mindsets of the people helps you to grow the type of bacteria in your mouth, gut, and on your body. On your body, wherever you have sweat glands, you have that problem. So that is a tremendous application in biology and human health. See, people doesn't understand. Okay, if I smell bad, what makes different? How many times you can brush? If you have a bad bacteria in your mouth, you cannot keep brushing your thing 100 times. Is it not? So we are also producing. Now anybody having ideas, you can also produce what you call mouth freshener, which activate good bacteria. So when see we made one such thing when I was in Panama. We made the student. We say first kiss to your wife. That means you don't even brush your teeth. If you are able to come nearer to a person, that means you have a good bacteria growing in your system. What happens if you have a bad smell? Anybody can predict? You are susceptible for anxiety. On a long term, you are very susceptible for anger behavior. And you are always susceptible for future Alzheimer's disease. So gut microbiome and also your microbiome in your mouth have a direct relation on your mental health. That's why Dr. Samuel Abraham explained on different conditions, Parkinson disease, autism, so many we have. So please maintain a good microbiome. And when you drink Coke, I always suggest drink a Coke because it's a fun. But drink a Coke only once in a month. If you want to know the impact of Coke, because some of us want to drink all the time, especially those who get stress, those who become anger, they want to drink because they want a sugar kick. So the important aspect is I will always tell children one experiment. Take a broken teeth, put in Coca-Cola. After 14 days, go and search for the teeth. Teeth disappeared. Teeth disappeared because it has acidic, highly acidic, which is 3 pH. You go and test your Coke, the pH is 3. And imagine I tell children, you take Coke, you have a dirty, your toilet, put the Coke. It's the best cleaner. Not even a small dot of things will be available. It cleans your toilet so nicely. Imagine what will happen to your intestine. All your microbiome disappeared. So all these things are coupled to each other, and there was a tremendous things Dr. Samuel Abraham explained. Okay, so please be aware of these facts. That's why where you need to find new ideas, where we need to produce good foods and good stimulating agents for your mouth, and all these things are very essential part. Okay, with this now we request you all to join for lunch. And after you come back from lunch, we'll have a sumptuous, most enlightening group discussions. We see who is going to be best idea winners, and then more than winners, we want to listen to the ideas and support some of you. You want to publish your ideas. That's the main theme we are having. So enjoy your good food. Okay, keep yourself more dynamically active. Don't get stressed so that you don't smell bad. Okay, be a sweet so that you produce amines. So be happy, be relaxed, and keep explaining us. Thank you very much.
Attention please. All the participants are requested to carry your ID cards for lunch. As well as you please complete your lunch within half an hour. As you are coming late, it is very difficult to hold us actually there. We are having some organization problem. So please complete your lunch within half an hour. Please move to lunch and complete it and come back and discuss. Please carry your ID cards while going to lunch.
Hello. Soumya. 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 Please report to the stage. Soumya. Soumya. Soumya garu, please come to the stage. Welcome, Mrs. Ravali. Our alumni, BTech completed. We were all more. Let's welcome K. Nitin Satya, the best volunteer onto the stage. Sahitya. Cheppaka. Sahitya garu, please come to the stage. Our alumni, KLU. All menu. Let's welcome K. Nitin Satya onto the dais. K. Nitin Satya. K. Nitin Satya onto the dais. Okay, booth la vachana okay. Soumya garu, please come to the stage. Amy. Copy కొట్టడానికి. Sahitya garu, please come to the stage. Poster presented by Sahitya Group. Veera Venkata. Let's play some songs. Hip hip hooray. Dhruti and the flying jet, please come to the stage. Nitin, please come to the stage. Please come to the stage. Nitin, please come to the stage. We request you to come to the stage. Yes. Huh? We see this Nitin backside. I'm gonna backside scope. Gayatri Garu, please come to the stage. Aman, please come to the stage for your poster presentation. It should be executed by 2 o'clock. Gayatri Garu, please come to the stage. ఓకే నించోండి నించుంటే పోస్టర్ ప్రెజెంటేషన్ ఇవ్వచ్చు మీరు ఓకేనా చెప్తున్నాను. ఏంటి
సార్ లాస్ట్ ఒక డైలాగ్
సాహిత్య అక్కడ రావాలి పైకి. ఎగిరిపోతుంది, పట్టుకోనమ్మా.
అది కొడుతున్నా.
చెప్పాను పైకి వస్తాను ఇంకా. అక్కడ ఏసీ పోయినట్టుందయ్యా. ఎక్స్ట్రా ఇది లాభం. చూడు.
ఆ లెక్క చెప్పినాము ఇప్పుడు. ఆ లెక్క వాళ్ళు లెక్క చెప్తారు. 520 175 పంచింది. మిగిలిన 50 పేపర్ ఏమేమి లెక్క చెప్పాలి.
సాహిత్య ప్లీజ్ ఫ్రంట్.
అక్కడ
ఏంటిదేనా బాబు? ఓయ్ పోస్టర్ ప్రెజెంటేషన్ పోస్టర్స్ తగ్గాయి కదా.
న్యూరో ఇంజనీరింగ్.
Participants, please be ready with your presentations. Vishnu. And we call upon the dice Aman from the AIMS Nagpur and Vishnu Reddy from the CSC, hey, BT second year. And the topic is bioinformatics and neurotech. Vishnu. Vishnu.
నీది క్లియర్ అవ్వట్ల పిక్చర్.
ఇంకా గట్టిగా
తెలుసోంది అబ్బా ప్రీ ప్రాసెసింగ్.
మైక్ ఆన్ చేసి. 10 please. Yes, sir. Okay. Spot prediction.
Uh, esteemed guests and dear participants, welcome back to the session. 5-day workshop, the 5th day. The last session we can say, and today is very interesting because all the poster presentation participants are very eagerly waiting for presenting their posters, and it is very much delighted to us to see all the posters and innovative ideas, the ideologies by the team members, and the few instructions before going to present the presentation, poster presentation. Number one, the participants are supposed to come together in the team. Say for example, say for instance, if the participants in the team are 6 members, all the 6 members are supposed to come onto the dais. So there is no excuse that one can present and other 5 participants can sit. All the participants are supposed to come onto the dais and give the presentation. And secondly, the time limit is for 10 minutes. The time limit for presentation is for 10 minutes. And once after the presentation, the followed by question and answer session. Followed by question and answer session, and the question and answer session is limited with 5 minutes. So altogether the presentation including the question and answers will be 15 minutes per team. 15 minutes per team, and the team members are requested to give in a nutshell the main theme of their idea because the time is limited, right? And I'm going to read out the teams now. The team number 1, yes, Sahitya, Anusha, Soumya and Chaitanya. They are not available right now. Okay. Okay, fine. Team number 2. Team number 2, Dr. M. Das, right? If I'm not wrong. Dr. Das. Yes, fine. And Miss Madhuri. Fine. Yeah, your team. Amrita. And team number 3, R. Nandini, Lakshmi Manasa, Jay Krishna, and Om Prakash. Right? All right. Next, team number 4, CH. If I spell wrong, please correct me. Bhavani. Right? Meghana, R. Meghana, and Kalyan. And team number 5, JV Kalyan, Srinivas, and Satvik. And next team is by Vishnu Reddy, Aman, and only 2 participants are given here in team number 6, if I am not wrong, right? And now, yes, all the teams wish you all the very best. And you know the participant with the name of Satvik. Anybody here? Yes, please. Please come and meet our Pro-Chancellor's ma'am, Pro-Chancellor's Secretary, Mrs. Shobha Rani. Hope all the participants are ready. Samuel. Hope all the participants are ready. Yes. And with a small change, actually, team 2 participants are requested to come onto the dais. And all the participants, let us give a big round of applause to the participants who are going to participate. And I request all the participants to be seated in the front row so that we capture the photos. And you also have the, your friends are going to present something here, so you can have a clear vision on that. Please move forward. Anyhow, you are also going to present now. Move forward.
Good afternoon, respected Jagannath Rao sir, the organizing team, and all the participants. Myself, Madhuri, a final year MBBS student studying in Pinnamne Siddhartha, Vijayawada. My teammates, Dr. Arunita ma'am, who is an assistant professor, Department of Physiology in AIMS Raipur, and Athira ma'am, second year postgraduate, AIMS Raipur. We are here to present a poster with the title Gut Microbiome Neuroplasticity Interface Model. During our fetal life, we acquire millions of bacteria while still we are in the mother's womb. Now, what is this microbiota doing inside our body? Like, even when two of us or three of us people sit together, we're not going to sit quiet. We talk. So, are the microbiota talking to each other or they're trying to talk with the body? This is where our curiosity began, and so we tried to identify various bacteria residing in the gut and how the gut is actually interconnected with the brain and affecting it. So, first, the gut and the brain are related by various mechanisms such as the enteric nervous system, which is present within the gut, the autonomic nervous system via the vagus nerve, the various endocrine hormones such as cortisol, serotonin, and other mechanisms. Also, there are certain specific such as immune mediators such as interleukins, which act as anti-inflammatory substances. Also, the BDNF, NMDA, and CREB. These establish an axis between the gut and the brain. Now, what is neuroplasticity? When an external or internal stimulus is given, the neurons reorganize into various structural, functional, and connections. They are modified. They can be either synaptic or neuronal. This modification occurs in every individual, which is normal. Now, we are here to understand in detail what is a normal gut microbiota and how it is influencing a normal individual. For this, we put up a research question, that is how normal gut microbiota play a role in neuroplasticity in normal population. But the knowledge we have about what is normal microbiota is very limited. So we cannot establish any relation to the neuroplasticity as of today. We would like to bridge this gap. Now let me introduce you to our plan of work. First, we are going to take a questionnaire from the subjects by their diet, age, gender, and various parameters, excluding people who have any chronic illnesses or known neurological diseases. Then, we're going to collect fecal samples from them using a smart capsule, maybe if it is available. That is, a smart capsule is something that once we take the capsule, it collects pieces where required based on the pH. That is, one side of the capsule has a pH monitor and the other side is for sample collection. That is, by reaching the appropriate site, it collects the sample automatically. Then we're going to analyze this sample, and then we're going to achieve we're going to classify the bacteria based on the diversity and the quantity. Then we're going to analyze which is the majority of the bacteria and the minority. Then we're going to take them through a series of proteomics and metabolomics. That is, what is it actually releasing? The byproducts and the proteins that are affecting the human. We are then going to okay, the bacteria is releasing a protein. Is it binding to the human protein to cause any effect? Then we're going to do a protein-protein network analysis. Then we're going to see if it is binding or passing through the blood-brain barrier. Then simultaneously, we're going to take an EEG to monitor slow delta waves which indicate neuroplasticity, and also an fMRI. Then we can also use some biomarkers for neuroplasticity, such as serum BDNF, small chain fatty acids, using gas chromatography and mass spectrometry, or eotaxin 1. Now, with this combined data about what the brain is doing for the neuroplasticity and the gut regarding the proteins, we're going to create an AI-based mathematical models. Then we shall be doing molecular dimension dynamic stimulation. With this, we in an artificial environment can understand how each one is interacting with each other. We then now we then will be able to understand which and how the normal gut microbiome contribute to the neuroplasticity. With this knowledge, by then we shall be able to give targeted nutrition to everyone. As India is a developing country, large population are below the poverty line. So a simple modification in the diet can lead to major impact. Also, in the children, if we catch them young, we can implement this diet through the midday meal scheme, which is ongoing. We can save a lot of amount and also provide healthy life to many people. Then it can also be not only be used as an probiotic. If we identify any pathology, we can use it as a nanoparticle technology and implement it in the treatment also. We have used these references for our poster presentation.
Which year these references were? Which year? Published when?
20.
I have three questions. I just want to ask. Now you have like series of microbes. How you are going to map them?
Okay, so
I
first,
just want to know technology you know about it so that you can beat the AI. You have 10,000 microbes. We cannot do by petri dish. There is a method where you can. There's something else. So it's a very simple instrument which can analyze series of microbes. Yeah, like in COVID, we did that analysis, nasal microbes. So it's called nanosequence. Okay, you take a sample of your fecal material or the fecal material or any one of the small blood sample or gastric thing or your nasal. So you can put and in a few hours time it do RNA sequencing. It will give all the bacteria that are there, all the microbes that are there in the human body. So that will give you enormous application part. My question is how it is going to change neuronal plasticity? Gut microbe, how it is going to change the neural plasticity?
Sir, actually,
Do you think it will change first?
it should change. We think it should change. That is our
Then
hypothesis
why
because,
and how?
sir, there are the proteins that are released. It has been found that some proteins from the gut microbiota or the components of the gut microbiota, they cross the blood-brain barrier. Not only they cross the blood-brain barrier, they also cause structural changes and features that can alter the blood-brain barrier. That is also disturbed in neurodegenerative disorders. There are some harmful chemicals released in the neurological disorders that affect the blood-brain barrier from the gut microbiota. So we are trying to find out the normal ones so that we can understand which normal components are crossing the blood-brain barrier and affecting the neuroplasticity in a better way. Because once we understand what is normally happening, then only we can understand what is the defect. If we identify any protein which is affecting, we can then cause a mutation in that gene and protein gene, and then we can check whether the same thing is responsible.
I want this girl to answer whole poster in 2 sentences.
Yes, sir. And I also want to add to that point, sir. Like we know that in eubiosis, the mycobacteria, the bacteria or the gut microbiota that is present in the body, that is having an anti-inflammatory role. Actually, it is decreasing the inflammatory components, and in turn, it keeps the microglia in a controlled stage so that it doesn't become overactive. But in a dysbiotic stage, what happens is that there is a imbalance between this inflammatory as well as anti-inflammatory the molecules, and there is an activation of the microglia at the level of the CNS, which can in turn result in inflammation of the cells in the like neurons in the CNS. So that's also a point.
Yeah, that's theoretical, but in a practical experience is different.
Okay.
But I want you to highlight whole your poster in 2 sentences. Most significant point you are talking about said. No introduction, nothing.
Yes, sir.
Only highlighting. He said this is going to be kickball for this poster. Only 2 sentences.
Okay, sir.
You want to create this, you want to develop this, or you want to solve this. You can tell any one of them.
Okay, sir. So unless we understand the normal physiology, what is happening in our body.
Only straight point.
Okay, sir.
Straight.
So the gut microbiome has a definitive role in neuroplasticity, and this neuroplasticity is found to be important in case of neurodegenerative disorders as well as in long-term memory and cognitive functions. So by studying
Okay. How many people agree with this argument? Any comments from anybody? If no comments, we agree. If comments, we have to wait.
Sir, I just have a comment on the word definitive role.
No, you argue with them, not with me.
Yeah. So I just have the comment on your usage of the term definitive role. Gut microbiome has a definitive role in neuroplasticity. In my limited knowledge, I still do not know a proper in vitro neuroplasticity model which can, by far and wide, concretely prove that this is happening because of gut microbiome. Hence, the next question is, what kind of model will you develop to prove it?
This will be actually an integrated model encompassing your EEG and biomarkers along with the samples analysis. No. Recruitment will be based on the exclusion inclusion criteria. Like, we will, the normal subjects that we will find out after the exclusion and inclusion, we will subject them. We will take multifactorial dimensions in neuroplasticity, the EEG and fMRI, the biomarkers as well as the proteins. And with these all, like what are the biomarkers and what are the proteins involved that cross the BBB, we are thinking to actually basically this workshop is a eye opener. Like if we would have thought individually, we would have come up to this level and stopped, or at this level and stopped. But once we have attended this workshop, we came to know that there are multifaceted dimensions to go beyond this. Like when we heard about the lectures on mathematical models, we came to know that with this we can actually create a model. At this stage we don't know, and individually we can't do it. So we will have to take the experts with us to develop this. This is not a single unit model, this is a multifactorial model.
Yeah.
My next question is, even based on the last talk we just heard together, right? Can you not incorporate a methodology where you do a healthy fecal microbiome transplantation in an in vitro model to significantly prove that the neuroplasticity happens or not?
Till what I have read, fecal transplantation or microbiota transplantation is not yet so much successful. It still is in the experimental stage. So our aim is different. Our aim is to means our aim basically is to go through the diet modifications initially and find out the specific protein, if any, which is involved in the neuroplasticity that can be given through diet, the protein component. That's why we are including in the meal programs, and if not possible, then as a probiotic. And like yesterday, we had seen molecular docking for the drug development. So we can use that model also. This is a multifactorial model. Like here also, when we have kept the protein dynamic simulation also here, so that when the protein we are thinking is crossing the blood brain barrier, so how it is crossing, what are the changes, we can simulate that, so that what we get is a good protein that we are talking about, that is helping in neuroplasticity.
You all agree, First thing is I should congratulate the team because they attentively understood the challenges discussed by all experts. Okay, they put all of them, all the ideas in one basket. The uniqueness of this project is they are not attempting to go into fecal material transplant. We're still far away, you know that. But what they were attempting to go is on the nutrition modifications. So that's one of the important parameters. And the second important, they touched a bold idea, I call it, because neural plasticity is still not known, even in normal. Even in my brain, I don't know. But they try to incorporate targeting like a moon. They want land in a moon using the technology. That's where, the ideas are really good. And I'm very happy that three medical doctors talking something different. If you'd asked them, they would have talked, they have spoken blood pressure, trauma, stroke, all kinds of things. But they went into a different direction. And I'm very positive that this can be one of the good proposal for your writing the review. We help you. Only thing is you three of you, because belonging to three different institutions are working, I can request, once you have a good strong idea, I can request Dr. Samuel Abraham to support, even he can be a co-author for you people. Good luck. Yeah, post it. You take a picture of the posters, huh?
You can place your posters here.
Okay. ಇದೈತಲ್ಲ ಜೊತೆ. Second one.
Group number 3,
Lakshmi Prasad.
R. Nandini, Lakshmi Prasanna, Jai Krishna, Om Prakash.
Even you do mistakes, don't worry. We want listen even mistakes also. Okay. Please. 10 minutes for presentation. Okay. No introduction, nothing. Start with the idea.
Good afternoon, everyone. So, our poster is on hypothesis model for treatment of localized brain tumor. It is on glioblastoma. It is about brain cancer. So, as we all know, it have become a great task to identify all the brain disorders, and of course, this 5 day of workshop is regarding this neurological disorders and all of the big brain. So, if you ask me, in this recent times, in this recent years, COVID have become a major task, and it have affected brains, especially brains. So, one of our closest people, one of our closest friends, have faced many issues with that, regarding many disorders like encephalitis, meningitis, and many doctors have couldn't recognize, like there was no symptoms and it was completely asymptomatic. So my our team's main motto is to identify, and it's like something hypothesis model to detect and to detect the brain cancer in another way, like stopping the ATP generation right from the glycolysis process. So if you ask me, why do you select that process and why not radiation and chemotherapy, radiotherapy and chemotherapy? It is because radiotherapy and chemotherapy have become also a process, but it has major disadvantages, like many people are undergoing weight loss, hair loss, and if you ask me, of course that is uh, it is very minimalistic and it is very small symptoms, but it is throwing a greater impact on people's life. So our main alternative idea for this is just terminating and inhibiting the ATP generation right from the glycolysis, like metabolic pathways are very important in a human's life, which will drive. Yeah, okay, sir. Okay. So this is the major outline of our poster presentation. So as we have already went on onto a workshop like in Brinda Hospital, a tumor usually. So usually whenever there is a development of tumor, it creates a pressure in nearby the brain cells that releases few symptoms like headache, appetite loss, and many other things like that. And in few situations it is also asymptomatic, and that will lead to encephalitis and meningitis and inflammation of brain areas and layers of the brain. So now one of my friend will explain the process of our hypothesis model.
Good afternoon, everyone. Actually, I got this idea from my intermediate itself. So actually, when I was in intermediate, I used to work on glycolysis cycle and ETC cycle. So mainly the cancer develops due to rapid cell division. Even that is not only in brain as well as in every part of the body. So for this cell division, we need rapid energy. That is nothing but ATP we are getting. So we have many drugs like they inhibit the tyrosine kinase. So these tyrosine kinase inhibitors are not specific, like only not specific target to the tissue where the cancer is developing. It is also inhibiting other sites also. So for that, now we came with a localized hypothesis where we can exactly target the tumor and without side effects, we can stop the growth of the tumor and we can cure it. Like we can inhibit the glycolysis pathway and we can inhibit the P2 receptors which are present on the astrocytes where the ATP goes and binds so the energy won't pass to the cell. So the cell won't divide. So this is our hypothesis. When like blood vessels supply glucose to the glial cells. In glial cells, we are discussing about the GBM, glioblastoma. Glioblastoma mainly caused in astrocytes. So when that glucose, if we inhibit the glucose to the astrocytes, it won't enter into the pathway of glycolysis, the basic pathway. So then the energy won't be released. Glial cells mainly depend on the ATP which is coming from glycolysis. So that is one of our point. We can stop the binding of the glucose. And the other point, the ATP which is already present in the bloodstream that goes and bind to the P2 receptors which are present on the astrocytes. If we block these two pathways and these two signaling mechanisms, the tumor won't grow, the cell division won't happen, and it can be reduced. And we have several ideas like how we can approach our hypothesis. Like we can go through nanotechnology where the nanoparticles goes and bind to the exact target site. So still we are in the learning stage and we want to work on this topic. And one more thing, like we have seen the neural navigation where exactly the tumor is present, so that we can get the coordinates of the where tumor is present, and we can use that technology to solve this. And we can also use some devices and sensors where the energy is tapping more in the brain tissue, because every tissue taps energy in a normal way, but whereas the cancer tissue it taps more energy. So we can create a device and place it in certain part of the brain, and we can get the coordinates where exactly more energy is tapping, so that we can create a drug with by using nanoparticles where exactly it goes and binds to those receptors, and we can inhibit the pathway so the tumor can be inhibited and we can cure it very easily. Because whereas other treatments we can see there are a lot of side effects, and we have only another pathway like we can identify the protein biomarkers and we can create monoclonal antibodies. This is one of the best pathway, but isolating CSF and identifying the a specific protein is very tough, and even we can do with that also. But our main motto is this part. When it is successful, it is not only applicable to only this glioblastoma. This pathway can be used to other cancers also. Now we welcome any suggestions and all.
Good. So his, conceptualization, he focused on many things, not on central point, one central point. So the one important aspect is cancer, GBS is one of the challenges even today. Diagnosis, cure. Both. Still we have a challenge and we lose the patients, honestly. His target, they were talking, is on the energy pathways. How localized system you are going to block, where you are going to block, because you cannot block glucose, honestly, because selective population of the cancer cells are there. So, and the cancer cell is a glucose loving cell, called, because they always take more and more and more glucose. So once you do it, energy will be there. Cell need that kind of thing. How you are going to block, if you want to block at glucose level, and if it is feasible, number one. Number two, if you want to block at electron transport chain level, where you want to block? You have 1, 2, 3. You know that ETC 1, 2, 3. Where you want to block?
Sir, actually, I have not gone into that depth. This idea just I got and I was developing it. So I have to know whether we can stop glucose binding site or not, or else if the glucose already entered into the cell, so it is processed and acetyl coenzyme is
What is
produced.
glucose binding site in brain cell?
Those things I have to start working. So just
Okay,
the I had
don't worry,
developed idea.
don't worry.
Okay.
So you want to target a transport chain 1, 2, 3. Where you want to target?
ETC
If you have
one.
an idea, otherwise don't worry.
I thought in the beginning of the ETC cycle we can stop, because in the beginning of the ETC cycle also we'll get the ATP throughout the ETC cycle. So at the cytochrome complex 1 we have to stop.
Why?
Why? Because already when the NADH goes and enters into the ETC cycle, in the initial complex itself we will get some ATP. So that's why we have to stop. And I have one more idea, like whether we can, we have to try it. We have to take a normal brain tissue and the cancerous brain tissue, and we have to try out all this, like whether we can inhibit the NADH or not in that particular target site. So I have to work on this very well. So I would request if there are any pathways or any approaches we can approach to solve this problem. We are mostly welcoming all those suggestions.
Because GBS is one of the very complex metabolically. Okay. So if you look at the oxidative parameters of a GBS, it's tremendously almost 5 times more than normal cell. So it consumes, your brain consumes 80% of your oxygen. Okay. Out of that, if you have a cancer cell, it consumes out of that 80% in the brain, it takes off almost 30 to 40% of oxygen. In your normal, compared normal cell, brain cells capture. So they are highly energetically active. And these cells are redox potential because you have electron transport chain, high transmitters happening. They're very active in the redox potential pathways. So that's why they cycle themselves, because they need to support energy in the transcription level, because they need to allow cell divisions. Lot of active cell divisions going on in the brain, in the tumor cells. That's why they expand very effectively. GBS has another problem. The cells try to capture surrounding cells also. They use that machinery like virus. They use that machinery to grow themselves. That's why GBS has become one of the very complex, challenging problem even today. Even today, because targeting directly radiation therapy or the chemotherapy in GBS has become a big challenge. And you know that once person has a GBS, invariably his life quality, death is almost within 3 months. We cannot prolong much because the rapid growth and also handling the complex issues many times, this happens. But it's a good, you're trying to put a lot of biochemical pathways in this. So if you put all the biochemical pathways in that, it is going to be tremendously impact. But if you read latest reviews on GBS in last few months, plenty of them are appearing. Plenty of them. People giving all kinds of activeness, microRNA based, because it has a multiple, biochemical pathways going on. Targeting one pathway is going to be very difficult in this process. But targeting your electron transport system 1 is very crucial, because you should know why.
Sure.
If you go through because it is the redox potentially active. Electron transport system 1 is the one where you have iron, ferrous ferric, Fenton reaction we call. So high energetic electron transport chains are going to happen there. If you block there, it will also block the Krebs cycle, because you have a FAD and NAD is coming into that pathway. Those mechanisms are very active, and people also trying to put low carbon dioxide, treatments into these cells. People are trying different methodologies. This is one of the important driven mind. I know you are biotechnology, still you need to understand a lot of biochemical pathways, but you made a good attempt. Okay. Any questions from any audience? Medical, non-medical, engineering group? If no questions, we congratulate you people. Okay.
Thank you, sir.
Please.
Actually, I have to thank our Pro Chancellor sir and Anita Mahadevan madam. So when I got this idea, I have approached her. I asked her whether I can do it or not. She said that you can do it if you are interested. You come to NIMHANS so that we can develop the process. I will help you, she said. So I have to thank her and sir.
Congratulations, and you go for your internship.
Sure, sir. Thank you, sir.
Group number 7, NDD. Group number 7, NDD.
Listen, everyone. All the participants are requested to fill the feedback form. It is mandatory for DST served workshops. It is requested, like, need to fill the feedback form. Please, we have to submit that.
You are presenting.
We as a team of
And I request all the poster presentation participants, you please post your poster as well as your presentation photo in the LinkedIn and tag KL University, please. All the participants who have presented your poster can take a photograph of your poster as well as your presentation photograph. Please upload in the LinkedIn and tag KLF Jagannath Rao Garu or Vasudha Devi, something like that. Please tag KLF. KL University is the official LinkedIn, that is KL University. Please tag.
So good afternoon, everyone. We are from Department of Biotechnology. So we all know that 75%. So actually, we did research on Alzheimer's disease. So 75% of the cases are caused due to amyloid protein. So what about the remaining 25%? So we made a research on the remaining 25%. So um, there are proteins like transtheraptin and TDP43. So we would say that they are probable biomarkers to detect neurological degenerative disorders. So let's take transtheraptin. So transtheraptin is a transport protein. It transports thyroxine and retinol and it is produced in the liver and choroid plexus in the brain. Transtheraptin, its function is to transport, but how it is related to Alzheimer's disease. So I would say that if there's a mutation in the so transtheraptin protein has 127 amino acids and it is a tetrameric protein. It is a tetrameric protein, and if the mutation occurs at the CD loop, it causes cardiac amyloidosis. So amyloidosis is a process where it causes amyloid proteins. So there is a research that has gone and they proved that transtheraptin will act as a neuroprotective protein. So it aggregates with the A beta proteins and it acts as a neuroprotective protein. I would tell that it doesn't act as a neuroprotective agent. See, these transtheraptin proteins, there was a research that they took the blood samples from the patients of England in year 1997 to 1999, and there was a follow-up till 22 years, till 2019. There were cases that decreased level of TTR, that means transtheraptin protein in the plasma or CSF, cerebrospinal fluid, it causes dementia, and later on from that it causes Alzheimer's disease. So why can't it be it can't be it be the reason to cause Alzheimer's disease? It can be the reason, right? So why can't we work on the transtheraptin protein as a neuroprotective agent? Neuroprotective protein, it can act as a neuroprotective protein, or it also may be the adverse cause to act Alzheimer's. There was 25% of the cases where there was an amyloid protein causing Alzheimer's disease. This may also be the cause, and TDP43 may also be the cause. So my teammate will tell you about the TDP43 protein.
Good evening, everyone. Sorry, good afternoon. I'm here to explain about the mechanism of the TDP 43 here. So, how it's going to lead to a Alzheimer disease. So, you're observing here, right? It's present in the nucleus of the TDP40, nucleus of the neuron. Here, it get present when so because it has the role to synthesize in the cytoplasm to get the sorry sir, I was not there.
It is a protein involved in DNA repair.
Okay.
Double strand repair. That's why it is present in nucleus. If it comes to cytoplasm, it is a problem. Go ahead.
So here amyloid precursor protein, it releases the amyloid beta monomer. It get evolved into it get transferred into the amyloid oligomer. So after that it goes to the fibrous and amyloid beta plaque. It get accumulated at there and aggregation. It due to the over contamination, like over concentration of the accumulation, it blocks everything at here. So the signals of the other neuron to this neuron get stopped. And this one, this one is microtubule covered with melanin sheet. Axon. The inside is the microtubule. So here it gets the block, and there is another way.
Covered by myelin sheath.
Sir, I think the microtubule should not get degraded into.
Simple. Axons always transmit electrical power. All neurotransmitters, the thing, they all have electrical signals, neurons. The neural signals should not go out.
Okay.
That's why you have like a wire, This myelin sheath covers that. That's why it's lipid.
So that means signal should not go outside the fluid, and so to hold the microtubule. Okay, thank you, sir. So at here there is another way to degrade at this place. That is TAU protein. That one get degraded at here for a longer time accumulation. So later, after the whole accumulation of the neuron, then this TDP43 gets need to act its action. So it get deplaced into the cytoplasm. It get deplaced into the cytoplasm. So it get accumulated at here. So, sir, how it's going to be act as a biomarker? The whole accumulation get over here. So we can assume it may be in the while you're taking the samples of the patients, you can check the whether this 4D sorry TDP43 is working it's or not. So if not it's working, so we can assume that it is a biomarker and it leads to the Alzheimer. Thank you.
You all agree, no? They did a good homework, but they studied a lot of things. That's a good point, I should congratulate the team for that job. But you read a lot of things. They are trying to understand. We know that. I will tell you two mistakes what they have done. Okay. First mistake, the boy was telling 80% of Alzheimer's is caused by amyloids. Correct? In the beginning, first sentence, he told that. Okay. I would like to tell you. See, when me and Dr. Anita, we were working, we have a genius, extraordinary man. He died. So we took his sample. He was 98 years. Okay. He has no memory loss. Extra normal. extraordinarily normal person, and then we dissected and start working on amyloid plaques, deposition. Huge amount of amyloid plaques in his brain. There's no memory loss, no cognitive dysfunction, perfectly normal, 98 years. And then we have people who have cognitive loss, memory loss, all these things did not present any amyloids at all. So we were making an argument at the time whether amyloid is a hallmark of neuropathology of Alzheimer's disease. Okay, even today that's not proved. No drugs are available because we try to do monomer, oligomer, all kinds of things, Still we have not seen a drug which will be targeting only them and to clear. The interesting point of this poster, they went beyond amyloid. They went into TDP-43, which is normally predominantly seen ALS. But TDP-43 is every one of us is a double strand repair, protein which will translocate. And I think 2008 or something, there's a paper came from Virginia Lee and her husband saying that TDP-43 they were able to see in the outside of the nucleus in the Alzheimer's disease. So they were kind of proposing this may be a role. The important hallmark of this poster is they're trying to put many proteins together to say that they're all causing. They're all causing together, not individually, because you don't know individually. Where even amyloids are there, you have TDP-43, you have many, many things happening around. Above all, we have large number of diabetic patients who are, we have always developed a memory loss, you know that. When you don't maintain your sugar level properly, you develop cognitive parameters and also you have a memory loss very early. Because what happens when your glucose level goes up in the body, there are medical doctors here we have, many proteins get glycosylated. We call it advanced glycosylation end products, AGE. Protein, like synuclein goes that, tau goes, all these proteins will go through those kind of parameters. So those are the additional factors they are going to play in neurodegeneration. So this is a very complex they have taken, and they try to understand a lot of biochemistry, we know, but they are still in the learning phase. So I don't find issue because they are learning phase. So they learn like we all learn. So that was a good job they have. Okay. If any one of you have any comments, I welcome. If no comments, we congratulate them. And then this also, if you put your energy learning a lot of biochemistry, parameters, we can request Dr. Muralidhar Hegde to help you people to write a short commentaries, on is Alzheimer's disease and essentially a protein risk disease. Something very catchy you can put and you can put across. But you have to learn a lot of biochemistry into that. If you people are really interested, we'll be happy to help you. Okay, congratulations.
Thank you, sir.
Group number 1.
On the posters, make sure you write your names.
Group number 1.
Names.
Saitya, Somya, group number 1. What's the time duration? 1, 2, 10 minutes. 1, 2, 3, start.
Thank you for having all of us here. So, our posture. So, our poster is mainly about this workshop. This workshop is all about the interdisciplinary. We are saying in our poster, interdisciplinary is strength in advancing the neuroscience disciplines. For example, how machine learning in all our four disciplines encourages neurosciences in their advancement. We are a group with different disciplines. For me, my name is Sahitya Puvada. I am from cognitive neuroscience background, and another of my teammate.
I'm Soumya. I'm from mostly medical devices and brain computer interface background.
I belong to a background of biotechnology and biochemistry.
I'm Anusha from AC department.
So basically, we see here how this machine learning advancing our cognitive neurosciences or the neurosciences disciplines. So here I'm talking about the cognitive neurosciences and how machine learning helping us to develop more. So in cognitive neurosciences, we have basically the inputs from all the sensory organs, which are like all the sensory organs have different encoded inputs in our brain. Specifically, we can help us to understand language, decision making, or motor controls, or language and memory and the perception. These are all the experiments we could go in this cognitive neuroscience discipline. So basically, what we do, we just take one type of sensory input, and we try to understand the mechanism in the brain itself. For example, myself, I take up machine learning and AI biomarkers for migraines. So I take visually induced migraines because there are some patterns in our world that we generally sensitize when we see specifically some odd patterns or the carpet patterns that directly induces migraine in our brain. So how the person habituate or sensitize, we don't know. So we generally take a data set of the healthy participant based on the ethical approvement of all the professors and the ethical committee from the institute, and we run a experimental designing using creating a stimulus based on the papers that are already published on the migraine informations. So I took a pattern-less stimulus, and I developed a data set with normal controls, and also I took people without migrainians but having a headache people. So I just cross compared during the data collection, and after the data collection we do pre-processing, and during the analysis part I do machine learning. So the machine learning is generally to understand the difference between the normal controls brain information with the migrainian people. So how the migrainian people sensitivity is different in occipital lobe. Specifically, we find this type of information in only in occipital lobes. So I found out that it's a large data set, and to process we use the information like machine learning tools like svm and mass univariate analysis. This type of analysis I have used in my work. So in that way machine learning helped me to progress in my work, because another people who are using say the same data set got a different resolution because of the advancements that they haven't used for machine learning. And my colleague will continue with.
And my domain is image processing. I take image processing, neuro image processing. In image processing, histopathological images are more accurate than compared to CT scan, MRI scan. In that, there is the steps: image segmentation, feature extraction, data integration. Then I take example omics, multidisciplinary omics. So in that, histopathological images are more accurately performed when compared to CT scan, MRI scan. So next, my colleague continue, Chaitanya.
So my interest is in integrating neuropathology with biochemistry. So my area of interest has been also like integrating MHC complex 1 Well, MHC complex 1 is known to make cells transparent for be it lymphocytes or monocytes, WBCs to act upon in case of an infection. And then we have tubulin associated units in dendritic cells. It so happens that tubulin associated units they degenerate. The degeneration of TIU is the primary reason for neurodegenerative disorders. And during neurological infections, like be it rabies or be it meningitis, it MHC complex 1 kicks into action. The uniting factor between both MHC complex 1 and TAU is that they both act on the cell membranes of the neurons or neuron cells. So a deeper understanding or the interrelation between the two can be done using molecular docking. So it's going to integrate both immunology and a neurodegenerative disorder. So it can result in a common ground between the two disciplines, because rabies, as Dr. Mahadevan had said on the first day, is incurable as of now without the vaccine. So it can pave a path to understand the brain better and the molecular interactions better.
Coming to the brain-computer interfaces, mostly we'll be dealing with the brain biopotentials. Where we'll be collecting, all the physically brain biopotentials and getting, done with our instrumentation part. So here, what we are trying to do is, once we acquire the data, we'll be, applying the pre-processing techniques, and then the feature extraction where we're using our AI/ML algorithms. From them, we'll be starting our validating our data with the existing data. So once we develop a validated algorithm, we start in this way. When we validate our algorithm with the existing technology, we'll be having a feedback loop, so where it will also get us the newly trained data as well, because we develop algorithm using bunch of online data set. That is how we'll do, because if we get the hospital data, we need to go through ethical clearance, where we don't have anything to show to them in the medical device market. So we'll be developing our algorithm with the online data set. Then with the ethical committee clearance, we'll be getting the approval and validating our algorithm with the online conventional EEG machines or conventional MRI machines and all. So once we validate it, there we start a feedback loop of our algorithm. So where it will be trained as a medical algorithm rather than just an algorithm technology, it will be in aspects of medical algorithm. So in that way, we'll be able to get the higher accuracy and a medical grade product rather than just saying a technology product. So as an engineer, we'll be mostly looking as a problem and solution. But when we comes into a engineering to medicine, so we need to think in a patient perspective, doctor perspective, and a technician perspective. So in this way, BCI helps us in BCI integration with AI helps us in getting the accurate medical algorithms as well as medical devices. So that's from our team. Thank you.
So the group has synthesized, all into one dish, where they would like to couple neuroimaging, other biochemical parameters, and other pathways together, because they want to synthesize application of AI and ML. Okay, that's what they would like to. So one of the important, they were also looking at the possibility of making a device, whether we can predict anything. So the one of the important aspects in brain breakthrough devices so far we have, one is stroke, because when a person has a stroke, he has his blood pressure in the brain, we call capillary pressure, goes up in the brain, and the capillaries start rupturing. That's how you get clot, and you get into problems. Sometimes you die, sometimes you get secondary issues. Okay, but if you can save a patient within that 20 minutes, not allowing a capillary thing to rupture is a big challenge. You cannot take MRI, you cannot do all those things. You don't have time. The Houston Methodist Research Institute, there is a neurosurgeon. He built what is called one carotid artery pressure. So as soon as patients arrive into hospital, the first thing they do is they calculate the carotid artery pressure, and they say this guy has to go for the surgery. They're only 20 minutes, remember. You have to make a hole, you have to make all these things. So what they do, they put a scanner which will identify where the pressure, and they use a laser beam to relax the capillaries. So capillaries don't rupture. So this is one of the most advanced technology the hospital is adapting in Houston Methodist, saving a lot of patients without undergoing any stroke condition. They save a lot of, and US has also a culture when somebody save you without any problems, there are large number of families donate their money and even their houses for the hospital. There's a culture there. A lot of people donate money for the research, for money, those kind of. So one such thinking device, I would like to ask these people. Okay, they are talking device, but I would like to know. I'm just gave an example so that you know what I'm thinking. Can you think of some device, even out of madness? Don't worry. But can you think of some device? You say, if I put this device, I know this guy has a Parkinson. He's going to Alzheimer's. He's going to whatnot. Can you think of that?
Yes. Yeah. Okay. So currently I'm dealing with epilepsy. So there, where we are not focusing on entire epilepsy, we are focusing on non-convulsive seizures and status epilepticus. I'll tell you brief you about status epilepticus. See, status epilepticus is condition what prolonged seizure attacks. It is you are getting the seizure frequently within 10 minutes, or you are getting the seizure continuous even after 5 minutes. So you're getting prolonged seizure attacks. This maybe like sudden unexpected death are very less, but coming to the outcome of these patients is you'll be into a comatose condition, you'll be having a memory issues, you'll be having an altered mental state every time. It is very frequent. So these are the quality of life where it is getting affected for these patients, non-convulsive seizures and status epilepticus condition. So for this, we want a device where it will tell like your seizure should be whatever seizure you're getting, it should be stopped or terminated within within this much time, so that it won't convert into a status epilepticus. Because detecting seizures, there are many home care devices or the medical EEG which is doing that. But coming to the non-convulsive seizures, where you will be not able to see any jerky movements. Generally, if you see a fits patients, we'll be seeing a jerky movements and all. But coming to non-convulsive, you'll be only there will be minimum to no outward clinical signs. So detecting that will need a compulsory medical device. So in that way, we are I won't be saying the whole development because it is under my patent. It is under process. But yeah, we are working on this to tell them like this can predict this can lead to status. Please take care. We're gonna take you're on a risk, so we are gonna do that.
Good. Actually, before I close this these people group, Apple Watch has built a precaution, heart attack issue, because many of us will be sleeping, we get heart attack. We don't know even. So many people die in sleep, that, because they don't know you have a heart attack. So they had a device so you can wear your thing, and it is connected to ambulance service. So sometime you'll be sleeping, you will get a heart attack, you will not be knowing, but ambulance will be in your house. So such module device is going to come in next 2 years time. I think we should be happy to see your device, and we really congratulate you for that effort. And those kind of modules we need to think of. Okay, thank you.
Sir, I'd like to add one more.
Please.
So using if we are able to get CSF analysis in a non-invasive method, we can go like a person above his 40 in his 40s can go through the treatment periodically, so that he could be well aware of his status, the state of his neurodegeneration.
There's
Or
no
the infection.
non-invasive for CSF. We have to use invasive method.
Yes, sir. That's what. Like for example, if we have a glucometer, so it using infrared rays, it does the prediction of amount of glucose. In a similar way, if you can do the CSF analysis of be it alpha synuclein or TAU, it would be better. Yes.
Good
But
effort.
yeah, that would require extensive knowledge
Yeah,
of
yeah.
physics as well. So
Thank you.
what the problem I found is, yeah, okay. One more problem statement which I'm dealing is intracranial pressure monitoring. So generally, this being done with the invasive technique, where they'll be inserting pressure sensor into your brain and getting the monitoring, which is a gold standard. You'll get the exact results of intracranial pressure. But the problem comes here is, as an engineer, what I'll think is every needle which is inserting is cost around 50 to 80,000. One needle, which is not a reusable, which can be used one time. But in our Indian scenario, off record, it is used many a times to save burden on the patient's record. So that is one thing. So but developing that pressure sensor, we have labs, we have experimenter, but the problem is I don't have a team. So this is an open call. If anyone wants, anyone have an interest on ICP monitoring, because ICP monitoring is being done. I did whole research. ICP monitoring is being done every day, at least 5 patients in a neuro hospital, neuro care. There are neuro hospitals, neuro care centers. See, 5 patients a day is a big challenge for us. As an engineer, I'll see that okay, every patient should get benefited with what I'm developing. So in this way, so we need to make that in such a way that pressure sensor, which cost around 50,000 to 80,000, to come down with the same accuracy. So that's my problem statement.
So we have some doctor team, you can think of, or you need any doctor team, we'll help you. Okay?
I have the doctor team.
Yeah.
I need expertise in
Oh, no worry. We have some teams in engineering. Some people are interested in devices they are developing. So we'll connect you. Okay? Thank you. Congratulations.
Group 5.
Next team.
JV Kalyan. Group 5.
Next team, please.
Okay. Good afternoon, everyone. We are a group of undergraduates here with our research idea. So this is the hypothesis that we made. It is an idea, and we need to test it. So yeah, I think I can make this our presentation more interesting. We are more curious or more interested in the sci-fi related movies. For example, let's take Matrix Reloaded, the Matrix trilogy. So brain-computer interfaces and what the person might think using a brain-related device. So that's the idea here. So it's been inspired from the movie perspective and also from the lecture from Dr. Srinivasa Chakravarti from IIT Madras. Okay, so our idea is like capturing the mental state imagery by using the EEG models. Okay, so the thing that we propose here is that we are trying to find what the person thinks in his mind by displaying some images. So we are from computer science background, and we are trying to imply some AI models by connecting it to the neuroscience field. Okay, I'll just give you the layman perspective of how we're gonna do this research. So yeah, the first thing is that we need a distilled environment. So EEG signals will mostly contain noise because a person might be thinking other thoughts while they have been showing some images. So okay, so we need to set up a cool environment and a calm environment, and then the person is being introduced to the images, like images of a single object, and he's been introduced that. And at the same time, simultaneously, the EEG signals are being captured. And after collecting this data, we'll then train a mode. First, we need to clean the data, like remove the noise and some outliers if there are any, and then we need to pre-process it for the modeling. And then after pre-processing and cleaning the data, it is then sent to the modeling phase. So here comes the power of AI that we are planning to use. There are some algorithms in artificial intelligence and machine learning, like the SVMs, support vector machines, and RNN2. So these help in classifying the sequential data, because the brain signals are the temporal data. We can see it through the temporal perspective, and time series data too. So then we'll try to apply these algorithms to the data, and we need to classify the dependent variable too. Like for example, if a person is shown only apple, like multiple images of apple, so then the signals are captured and model is trained. So afterwards, we are going to the testing phase. So in the testing phase, a new person, not the new person exactly, a person comes, and then we'll be setting up the EEG equipment to his head, and then we try to capture the signal. So if the model can predict if he's thinking some apple in the mind, then in the monitor we can get to see what he's thinking. So then we can see how model has worked. So this is the whole layman perspective idea, because we are coming from the computer science background and we are inspired and more curious to create that one. Yeah. Coming to the conclusion and applications, you know, paralysis patients, they are like really unable to express their thoughts. So we are thinking if this idea works, then we can try to find what the person like paralysis or disabled people can think, and it would be like a bridge of communication between the normal people and the disabled people. So this is our idea, and I hope everyone gets it, and I guess it might be interesting for you too. Thank you.
It's good, an interesting paper, where they are putting computational science, in mapping one of the important parameters of the brain problems, using the EEG as a parameters for them. How many of you know about EEG? Any idea? Many doctors are here, other people. You know that when you have a sleep disturbances, when you have a dream patterns, you have both positive and negative dreams. You know that. Some dreams are very sweet. Some dreams will be horror for you, and you have intermittent sleep problems. Like you sleep sometime, you get up, then you go to sleep, and then these are the major factors which help you to understand disorders now. See, for example, people are taking your sleep pattern itself for to understand your mental health issues. If you don't have a good sleep, you have intermittent sleep, you have a dream pattern. Dream biology is a big subject, right? Is a very big subject now. So those are the important aspect people are trying. So EEG pattern accumulating to one disease is very difficult, because they are overlapping. You know that either Alzheimer's, Parkinson, related things. People are trying to understand these parameters, and this group is trying to take out the differences and try to put them in order, so that whether you can using algorithms, whether they can build those kind of things. And I'll be very positive, that Srinivas Chakravarti will be happy to support with, if you go with a very clear data, either related to sleep. Sleep is one important problem. We can get you data from a brain check center we have in Dubai. Like, we have a brain check center in the Canadian University there. So we have a brain check center we established. I'm advisor there. So we can help you people to go a clear training, either here or the universities in Texas and other places. So if you take the clear data, then we can connect you to Srinivas Chakravarti, where you people can go and work 2 months, 3 months, and put an application part. Because as students, you always think one level of algorithm, but you should think beyond. That's where Dr. Srinivas Chakravarti or Sham Divakar will be helpful. Good luck.
Sure. Thank you.
Group 4, Kalyan.
Good afternoon, all. I have a quick question. How could we define health? Like I was previously listening to expert talk from BM Hegde. He's a doctor. So he gave a question, can anyone describe what is health? Then he answered that enthusiasm to work. As far as I am experienced, I used to get disease soon after seeing the school, and my disease used to disappear soon after seeing the doctor. And all this is due to placebo, what I feel. And as far as I know, healing is all about what we believe. And uh, I want to see into the future, so I have looked back into the ancient wisdom too. Then I came to the concept of Purusha and Prakriti, where Purusha is all about awareness and consciousness, what we are aware of and what we are conscious about. And Prakriti is all about creative potential. And let us break it down. When we go to the molecular level, our cells know what are invasive species, what are foreign objects, so they identify them and they kick them out or they digest using some enzymes. That is Purusha, because our cells are aware of what they are and what are invasive species. And creative potential is the genetic material by which the cells are formed. Uh, Previously, I was looking into a research paper by Joe Dispenza, who is the author of Power of Subconscious Mind. There, he was discussing about mental visualization. Uh, Before doing any task, before performing any task, I used to practice myself mentally so that the efficiency would increase. So, what I suggest is we can use mental visualization, including proper diet and also intermittent fasting, so that we can cure these neurodegenerative disorders. And the second thing is resonating frequency, where generally when a cell gets some disorder or disease, its frequency is way different from the frequency that are emitted by other cells. What we can do is we can create a set of music which aligns to the frequency of the disease cell, and then we can resonate them, we can change the frequency of those cells, so it can heal them. I mean, they can heal themselves. uh, as far as these 5 days are concerned, I've learned many things, like how experts from different fields can collaborate together. I'm from ANDS background, artificial intelligence data science background. So it was like a Malayalam film watching about neuroscience and all. But I've learned many things: what are biomarkers, how proteins and different metabolites can be used as biomarkers and all. Uh, what I want to contribute is we have to look at that ancient wisdom so that we'll know who we are, who our ancestors are, and how they dealt with the problems, so that we can deal with modern problems too. Thank you. And completely this chart is made by our team members. I've just gave them an idea and they've executed it.
It's fascinating, somebody with a computer background, artificial intelligence, trying to understand something, beyond knowledge of them actually. Where they were discussing about the role of music, influencing the brain, putting across how they are going to incorporate that knowledge into understanding the complexity of brain, like consciousness, subconsciousness, and then music, application of music. If you do into research, I know they have not done much into research because they are listening all of the things here. If you look at the research part, music therapy has become one of the tremendous applications. People, two things: people playing music, people listening music. We have two aspects. People who use like playing music, the pattern of brain response is different than the people who are listening music. Okay, actually, we are working with a brain check center where Pavani is going to involve. Like words you use, different words. Somebody use the word in anger, same word. Somebody used with an affection, same word. Somebody used in love. But word is same. Word is same, but makes a different meaning because you are using under different circumstances of the brain. When you use your brain, your easy pattern get changed. So metaphor we call, when metaphor changes happens, then you can see the pattern. That means one word makes differences. In the same way, in music, for example, all of us are listening. If I put all easy pattern for all of you, and if I take a same room, all the people talking, listening music, everyone brain responding differently. All of frequency is there, everything is there, but you start seeing the variation. So one of the studies have been done in NIMHANS, a few years back, where the people started putting Vedic chant mantras, chanting, and when people chanting, they were taking the easy pattern response. Because many times when you do Siddhi Yoga and other thing, suppose you have to reach a non-thinking situation. That means you don't think at all. That means your brain wave is straight line. But the people realized that you say I have reached that yoga, but they are not able to reach that yoga. People were doing those experiments in NIMHANS, and people were doing, and we also have one controversy, but of course we got a legal opinion because one your Siddhi from Himalaya came and said, I will go into Samadhi, record my brain. But legally there is a issue because something happens. It's a campus, but we built Samadhi for him, but we pumped oxygen. We make sure whether he takes it, doesn't take is different. But we found that he has a intermittent breaks of breathing. So those are the very crucial, which this group trying to explain, because they didn't have much time. I know if we give them 10 days time, I think they would have come out with number of those things. But it's a good effort you made. Now you have to come up with a synthesis. What next is a major challenge that is missing in this chart. Okay, good.
Group number 6, Vishnu and Aman. Yes, sir. Group number 6, Vishnu and Aman. Group number 6.
Hello. Good afternoon to you all. My name is Aman Milan Bhosle. I am a fourth year MBBS student.
Good afternoon all. I am Vishnu from Biotechnology second year from KLU.
So here we are presenting our posters in two parts. I would be majorly dealing with the applications of regression analysis in the patients in finding differences between the post-mortem brain samples of patients with Alzheimer's dementia and the normal population.
I am going to present the evolution of brain surgery technology in 1884 to 2040.
So, do you all know about a data set which is freely available and you can research on it anytime you want? So, due to the present technology, we have been blessed with some of the really good data sets which are available on a website called as Gene Expression Omnibus, that is GEO. And in that, they have data from various studies which have been conducted all over the world. So we have just here analyzed one of the studies in with our way of interpretation. So here we have taken a sample of 60 people, and their post-mortem brains were divided into 7 areas, and from those specific areas we have taken the brain sample, and then it was analyzed for RNA expression and the data was normalized. After that, we categorized all of the data, which was a really humongous data of 22,000 genes and 17 brain areas, into specific brain parts. And after that, we run a regression analysis for each of the gene, compensating for age, sex, and many other clinical parameters. And the most important part would be the diagnosis of Alzheimer's dementia. So we just did all this regression analysis, and this is the pictorial representation which we have made from it. I'm really sorry for the quality, but if anybody wants, then we have the online copy of it. So yeah, this is our project. So uh, so the only thing which I wanted to emphasize over here is being from a medical background doesn't mean that you cannot or you need not know statistics or anything related to that, because this analysis has been performed by us. Basically, I am not from a medical background, but still I am really interested in it. So I went, and the major thing which I want to emphasize over here is I don't know coding, but still I did it with the use of coding. But how? The thing is, I have used ChatGPT as a tool to code for myself, right? So this is a really emerging uh, field of interest for many of the youngsters, because even if I don't know coding, I can do coding right now, right? And I can do 20,000 regression analysis like within a span of 2 or 3 minutes. So that is a really impactful thing, which at least is according to me. So I hand over the mic to my friend for his presentation.
Uh, Actually, we know that the brain surgery starts from the 1884 for the Rickman and Goldie. It was at that time the brain surgery was very risky, and the success rate was very low. By using the basic tools like scrapels and forceps. And coming to the 1980, the technology was evolved. So we get MRIs, and we are little bit advanced. We are using, but the success rate was a bit high, not much. And coming to 2019, previous session, we have a session with Dr. Srinivas Reddy sir. He showed his innovation, it was very inspiring. Actually, my field is from biomedical engineering. I'm very interested in that. By seeing the sir's session, and I'm thinking of to collaborate with IoT and EC and biotechnology. And now we are using simulation in the 2040, like brain surgery, uh, medical technology. And in that, we are using the technology for the simulating, just for learning and training. In 2040, we are going to do that in the humans. So I do not have a problem statement in that, but I'm going to find out it. So it's also.
So they were putting two divisions, One is using algorithms, especially medical coding, is one of the hot area right now. Both for the even doctors have to learn now. Because they're going to use a lot of application part. You don't need to have every time an engineer next to you, For certain things, the medical coding is a very essential part. And also we are transforming many of the computer science guys also to get into medical coding, where they are essential in the biological system. And that one part he has used a database from a open access, and try to understand to identify the regression parameters. Okay, the other part he explained about the challenges associated from the way back, when WHO started, how the neurosurgery, risk to safe. It went through a journey. Okay, so that's a good part, but more work can be done on this line because we have a limited time. So more work can be done so that more answers can be captured. Okay, thank you. I would like to be, now we are going through posters, to evaluate thing. There's nothing like first, second, third. The best three people will get the best outstanding poster award. Okay, and then the important aspect I would like to highlight one more time. In the day one, when we started, we were always telling that you have to central focus on something unusual. Something, you talk something which other people are not talking. Even if it is a mess, we don't worry. We don't want a regular abstract presentations. That's why we want to see people come up with something new ideas. Now we can capture almost 3 reviews from these presentations. So we can say if they are interested, they spend time and they focus, we can really make 3 presentations on that. Okay, so we can connect you to Samuel Abraham, we can connect you to Muralidhar Hegde, we can connect to Dr. Chakravarti, Srinivas Chakravarti from IIT Chennai. We can work on those process. So it's a very interesting part. And when we were judging, if any one of you want to sing a song, because somebody said on music, somebody said on things, If anyone like to sing a song, I'll be happy to listen. So you don't get bored when we are doing our job. Anybody? No singers here? No language restriction. You can sing a song in Tamil, Kannada, whatever, Telugu, Hindi. Please, madam.
No.
Hello, everyone. I raised my hand because of my impulsiveness, but you need to adjust because I forgot that I'm having cold and cough. Okay. I'm a research scholar from Department of Biotechnology. My name is Tejasvi. So I'm going to sing Ramadas Kirtana, Yennaganu Rama Bhajana. Okay. ఎన్నగాను రామ భజన కన్నమీకిలున్నదా ఎన్నగాను రామ భజన కన్నమీకిలున్నదా ఎన్నగాను రామ భజన కన్నమీకిలున్నదా ఎన్నగాను రామ భజన ఆ సన్నుతించి శ్రీరామ సన్నుతించి శ్రీరామ చంద్రుదలచవే మనస సన్నుతించి శ్రీరామ చంద్రుదలచవే మనస సన్నుతించి శ్రీరామ చంద్రుదలచవే మనస కన్న పిన్నవారి వేడు కొన్ననేమి ఫలము మనస కన్న పిన్నవారి వేడు కొన్ననేమి ఫలము మనస ఎన్నగాను రామ భజన ఆ రామచిలుకనొకటి పెంచి రామచిలుకనొకటి పెంచి ప్రేమ మాటలాడ నేర్పి రామచిలుకనొకటి పెంచి ప్రేమ మాటలాడ నేర్పి రామ రామ రామయనుచు రామ రామ రామయనుచు రామ రామ రామాయనుచు రమణీయోకతే ప్రేమ మీర భద్రాద్రి ప్రేమ మీర భద్రాద్రి దాముడైన రామ విభుడు ప్రేమ మీర భద్రాద్రి దాముడైన రామ సగలేదా కామితార్థ ఫలములిచి కైవల్యమును సగలేదా ఎన్నగాను రామ భజన కన్న మీకెనున్నదా ఎన్నగాను రామ భజన థ్యాంక్ యూ సో మచ్.
ఓకే అనౌన్స్మెంట్ చేయాలి రీసెర్చ్ ప్రపోజల్
తను ఒక ప్రపోజల్ చెప్తాను.
థ్యాంక్ యూ ఎవ్రీ
Okay.
I just have a small announcement for everyone here. So I'm working on a research assistant as a for a topic on aphantasia, and here the aphantasia is about the people who do not have a visual image or imagination. So people who are interested in this topic, like we are going to understand the cross comparison study with the normal adults or the control people with the aphantasians to understand the emotional effects or the emotional realm with the aphantasia patients and how they are lacking with other cognitive abilities or how they are hyperactive with other cognitive abilities. So for that, if you are anyone interested in the aphantasia studies, or if you are having a doubt that your visual imagination is very less compared to other, or if you have any queries that you are in the spectrum of the visual imagery person, please work on the ice mind organizations. We are taking a survey actively from all the participants around the world online, so please join into the study if you are anyone are interested. Thank you. It's ice mind organization in University of Exeter that was conducted by Dr. Fraser Milton. Thank you.
Hello. So today I would like to sing a Hindi song. So Hindi songs कोई अभी सुनता है? Okay, that's really cool. Okay, so song goes like this. Hmm. Hmm. इन दिनों दिल मेरा मुझसे है कह रहा तू ख्वाब सजा तू जी ले जरा है तुझे भी इजाजत कर ले तो भी मोहब्बत है तुझे भी इजाजत कर ले तो भी मोहब्बत बेरंग सी है बड़ी जिंदगी कुछ रंग तो भरू आंखों के प्यालों से पीता रहूं उसके चेहरे का नूर बेरंग सी है बड़ी जिंदगी कुछ रंग तो भरू आंखों से प्यालों से पीता रहूं उसके चेहरे का नूर जब मिले थोड़ी फुर्सत जब मिले थोड़ी फुर्सत मुझसे कर लूं मैं हसरत है तुझे भी इजाजत कर ले तो भी मोहब्बत थैंक यू सो मच
We request some of you to provide your feedback,
Oral feedback.
whatever you feel and what suggestions you want. or what future workshop we would like. Before you start, I would just want to tell you, see, we have submitted a proposal to International Brain Research Organization on translation neuroscience. Okay. The selection of the candidates for the travel awards will be done by IBRO, International Brain Research Organization, not by us. So once it opens, we will be connecting all of you. We want you to people apply. So they will choose. They will only send the names. So that covers your accommodation, food, and your travel award by them. Okay. Other people can attend on a payment basis. This is going to be done in collaboration with the AIMS, Mangalagiri, and also the hospitals in other places where you will experience like full one day on MRI, for example. So one of the participant will be a patient. So you'll go through an MRI scan. So rest of the people watch your MRI scans, how they are going to do and other thing. And we do a lot of other easy pattern, other things. So where we put not only medical, engineering, and also other groups like psychotherapy and other things we will put. So we'll give an exposure what is going on. The second part, from this group, we will be watching, how active somebody. And then we have an Indo-France workshop coming up in February 2024, 19th February. So we are waiting for the confirmation of Dr. Daider, the Nobel laureate from France. Okay. So that is purely 20 people coming from France. All the top leaders in this country, IIT directors, AIMS director, Chandigarh, all kinds of top people, we are bringing them all together to see that we can build AI in medicine. That is a center for excellency we would like to create. And especially, I am suggesting, whenever we have discussion, please, either faculty or a PhD student or even undergraduates or a master students, please take the advantage of visiting different places to get exposure. Like somebody, faculty, those who are going to be there, if you have a good publication plan yourself. Not just you can apply because you are not going to be selected. If you plan very well, then you can, we will help you even, nominate you for the Indo-France Women Scholarship Program, where you visit the place for 4 months. And you should be tremendously active because they're mainly looking for life sciences, application part, and many things. So if we want to get you a recommendation letter, a nomination letter from France, they look at your CV, they look at your background, they look at your publication, so on and so forth. So please prepare well. So you plan next year. This year is already over. Plan next year. Plan now. Plan now. And also we'll be happy to collaborate with any one of your institution. If you want to conduct something in your place, we tell you how to get money from DST, how we can help you to get the people to you. All those networks will help you. So choice is yours. You need to build. You need to build. That's your career. You know that. So build yourself into a very strong leaders. That makes you, happy and healthy for you. So please try to put your ideas, try to put your knowledge, try to take the opportunities. And then you have a lot number of opportunity to go out and experience. You're not traveling abroad because you want travel. You travel because you want to experience what is going on in the world, how a lab looks there, how they are working. Like those experiences you need to have. Then that makes you wise and wealthy, which you also make money by going abroad. The second, it also makes you active. Okay. Keep these points in mind and be active. Okay. So you have any suggestions, you have any appreciations, you have any questions, you have anything, please feel free to put in terms of your words here. And in the meantime, we are finalizing your post awards. Okay.
Good afternoon. Hello. Good afternoon, everyone. Good afternoon, everyone. Okay, I just want to share a small message from my life and my journey in KL University and this entire workshop. I have seen very ignited minds working on different problems and different diseases as well. They are trying to innovate something in a different way. So, as a mediocre student and as a mediocre human being, I just want to convey a small message. So, when I was born with a defect. So, the defect is just it is on my left hand. So many of them pointed out me and what is that? Everyone pointed out me and asking what is that? What is that? I couldn't able to answer them what it is exactly. So then slowly when I'm growing up, you know how much pain will I get when gets swells? When a woman get period cramps, how much pain you suffer? Into 1,000 times I can say, because I have seen that instrument how it makes pain. So I have compared that pain to my pain. Into 1,000 times I can say. During that time I can't even able to move my hand, just I can't make my hand to lie on my bed as well. So when I was around 6th or 7th standard, so a huge pain, like I can't even express that pain. So I was sleeping during night time at around 1 o'clock, I guess so. I wake up my mother. I told her that I'm just getting a pain on my left hand. So then she said that why are you disturbing my sleep? Go and sleep. Just take this balm and go away. Then I went to my father. He said like, we'll see tomorrow. But they don't know how much pain I'm suffering with that. So then when I went to my grandmother, she took me and she saw that. And the next day we went for the MRI scanning. And it is very complex. Like the tissue is combined with the nerves. So that have taught me to get into science very more and more. Then onwards I have started spending my life with animals, plants and all those. I used to speak very less with the people. I used to spend most of my time in the garden or with the animals. So then I saw that MRI scannings, all these and those, and I thought that it might be a cancer. Then the doctors tested this and they said it is not a cancer. Then a thought came in my mind. So it is a small tissue. I was suffering with this much pain. Then what if a cancer patient, a huge lump of tissue is growing inside his body? How much pain he is suffering from? Then I thought that I should become a doctor. Then I should deal this cancer or whatever the disease the patients are suffering from. Then I tried very much. During my intermediate, on my wall, Rangaya Medical College is my favorite college. So I have written on A4 sheet RMC and pasted in front of my bed. So I tried to achieve that, but I didn't get into that due to some disturbances at the time. So then slowly, gradually, with the suggestion of my lectures, I came into biotechnology and I have started my preparation. But in this university, like not only in this university, most of the private universities are somewhere else. The student want to invent something in a different way, but that support from the management is very low. When the management supports such kind of students, they can innovate something in a different way. Like I can see here, Soumya and Meghana and you people are really good because you are working on different problems. You want to serve the society in a different way. So many people think that India can't do anything, and they think that only we have to go to abroad and we have to achieve something else in a different way. Do you know in IIT Kharagpur or Kanpur, I guess so, they have developed a device through the retina we can test the sugar levels, blood sugar levels. So it is happening in India, not there in somewhere else. So when you think you can do it in India, you can do it because I have a quotation just now. I have read that ISRO director have said this: science originated from Vedas, but repackaged as Western discoveries. This is exactly true because when you refer Vedas, you see what are the problem you are having. For each and every problem, we have solution in Vedas. Now, what the Western country people are doing, they're picking one point from Vedas. They are telling that it is a research discovery. We have done this research. Because when we say Vedas are very true, we don't believe. We ask for scientific proof. Am I correct or not? When a patient goes to doctor and doctor says something, this is this, some patients will really ask, is there any scientific proof? Can you tell me? So we as a Indian, we have to believe ourselves and we have to achieve many more things. Here, many more people are targeting different things. And one of my friend is working on stem cell, and I can see many more people are working in a different aspects. And a AIM student, he is interested in neuroscience. He want to do something in a different way. So I request everyone to choose your domain and try to target that domain. Get something message from the society. Try to solve those problems. And Chandrayaan 3, how many of you know? I don't know. Chandrayaan 3 is the project. Chandrayaan mission started in 2008. Many countries fought for achieving that mission because landing in the South Pole is very dangerous and it is completely dark. So when Japan is more advanced in technology, more than India. So for example, now we are living in 2020. Japan is now living in 2060. So even they also failed. Russia failed. Just 2 days back, Russia failed. So we have achieved it because a teamwork. The best minds are in India, not in abroad. You can see the top companies. They are from India. So she's, I don't know, Soumya, she's really doing a very good job. So don't think that's okay. She's really doing good job. The best mind in India. So if she invented something like a different device, where it goes, it enters into the abroad market. So suddenly if you ran away into the abroad and if you are inventing those things at there, they will say that these are Western inventions. They won't say that Indian inventions. These are the Indian people we did. They say that this is our company work or our institute work. So we have to cross all those and we have to make India to develop in a different way. So for that, each and every student need to collaborate and target on their particular domain and try to invent new things. And as being a selfish human being, just we are focusing on the technology and the development. Just we have to focus on the environment as well. Because you can see July 3 and 4, the average temperature of the earth is 17.6 degrees centigrade, which is very huge. Actually, it should be like 11 or 12. See how many degrees it has raised. So we have to focus on the environment as well. Because now within 5 years, not only India, each and every country is going to face different environmental issues. All the diseases or whatever the difficulties we are facing are due to environmental issues. So I request everyone to focus on environment as well. And I request KL University to conduct a workshop on environmental issues so that people can come to know about what is happening with the environment to the society or with the globe.
Sure, ma'am. Thank you.
And thank you. And like when Chandrayaan 3 mission is landing on the moon, we have seen the video here. But when the flag is flying on earth, we are singing Janagamana. When it is flying on moon, we didn't sing. I request at the end, please try to put Janagamana.
Sure, ma'am.
Thank you.
I request other participants to give your feedback, especially from the outside of KL University. Here, medicos are there, physiologists are there. Please, can everyone can give the feedback?
So good afternoon, one and all. I'm Dr. Nazia, Associate Professor, Rangara Medical College. So I happened to come to know about this workshop through my friend, Dr. Uma, Biochemistry Department. First of all, it was a very new thing for me. First time I'm seeing this interaction between the engineering and also the medical domains. It was really an eye-opener, and I've seen very brilliant young minds opening up their creativity and putting up into the execution, as already Jay has told about them. And I really appreciate Jay for giving such a beautiful talk. It was really nice and inspiring from young minds like you. And we would also like to invite and collaborate with the KLU and invite for some beautiful lectures from the KLU on different departments, especially AI, because this is very new to all of us in the medical fraternity. It's quite new, and we would like to open a platform for them to come to us and collaborate with our institute. And I hope so that this is going to be a great breakthrough in total science and technology department wise. And coming forward, the Chandrayaan 3. So we're all proud that the Chandrayaan 3 has made to the moon. So let us all make up to it. Thank you.
I request travel awardees to come and share your experience over here. Some of you have taken travel award. Please come and give your feedback, please. It is to be recorded, actually. Thank you. Ma water. I request all the travel awards to be present here to take a photograph, please. Travel awardees. Travel awardees. Please come.
Actually, we gave a travel award, as supported by DST, which covers, like 3,500 rupees, for each person. And then we actually awarded 10, but some people could not come because of the distance and other thing, and also timings. So, but anyhow, we have great appreciation because we have these awardees, which has won out of the competition. And this is, we have taken the consideration of their write-up, their CVs, their backgrounds, also their institutional backgrounds and their specializations. So we want to distribute in different angles, starting from computer engineer to doctor to other groups. We have taken all those things into consideration and did that. So as I said, the IBRO awards, when we conduct another program, it will all will be done by IBRO. You have to apply online, nothing like offline. Okay, so keep that in mind, and we congratulate all the awardees here. We have all the way traveled, in a short notice. We know that we have a short notice, but they came, and we are very positive that we could get lot of teamwork, from the different institutions, starting from Chennai to up to Delhi, AIMS and everybody. And the opportunity, we have chosen the speakers only because who are going to collaborate, who would like to help the participants, not somebody who come and give a speech and go away. That's the kind of mindset we have. That's where we built this process. So I congratulate all the travel awards for excellent job. Okay, so Randy.
Yeah, sorry again, but I'll take a few minutes to thank for conducting this workshop. My journey started with Vasuja ma'am in Delhi, where we are attending a international brain research organization, computational neuroscience and AI. So that's a 5-day workshop where we have met. So we attended, we got a similar experience as this, where we have undergone all the how the computational neuroscience and AI is being integrated to in more research works and all. So there I met Dr. Vasuja ma'am. I really want to thank her for giving me this opportunity to be a part of this wonderful workshop. And Sir Jagannadhara, sir, thanks a lot for being an inspiration for us. We were seeing every day you are sitting there and listening to all the lectures and the questions, and you are giving the feedback. Even you are listening my question is which I'm throwing a lot. So has been a great experience for me. It's been an inspiration for me because my background comes from a bachelor's. I'm just a bachelor's. I graduated in 2020. So it's been just 2 years where I started my journey in AI, but I started my journey in medical electronics from past 4 years. But this AI has given me a huge hope that I can survive in this particular medical domain where I can serve people as an engineer, where I can design a devices where it can help them to reach a better management or better diagnostic techniques, where it can help even give the clinicians informed decisions. So that's where my journey is going on, and this workshop has really helped me in networking, getting knowledge and getting new friends as well. So yeah, that's totally and kind of overwhelming for me. So thanks a lot, thank you so much.
Thank you, Soumya. Yes, delegates and participants, really it's a wonderful day because from the past 4 days, including of today, the 5th day workshop has come out with a wonderful result in the form of poster presentations, where we are very happy and glad to see a number of thoughts, innumerable ideologies with multifaceted by using the multidisciplinary aspects. So as our Pro Chancellor sir always guide us by saying that explore yourself, explore, do visit and have the connectivity, international connectivity worldwide, so that we can when we explore ourselves, then definitely we'll come to know a number of new things. So that was proven practically with today's poster presentation, and all the poster presentations which were presented by the participants were really extraordinary, thought-provoking and brainstorming. So once again, let's have a big round of applause to all the participants, by all the participants, for their active participation and wonderful presentation on this auspicious day. And one more thing, it was very notable and remarkable that all the participants were very keen and meticulous in observing the presenters. Say, for example, Professor M. Srinivas Chakravarti sirs and another keynote speakers' presentations. Keeping in view of those key points, they started off working on that, and they started thinking and they started doing. So it's really a great success. So I really, on this platform, I assure you that the participants of this workshop, 5-day workshop, are definitely going to carry valuable inputs, and definitely you are going to do something definitely wonderfully in future. And definitely we are proudly, definitely we'll say on some of the stages on the other day, these are our participants of our so and so conference and workshop. Really, yes or no? Yes, thank you for that. And out of all the poster presenters, as you know, very marginal, very 0.1 or 0.2 point, not differences, but you know, all of you, first, second, third out of all can be first, second, third only, because that is like I can say very minute, not difference. All are winners. As Pro Chancellor sir said to me that all are winners. All are winners, but only for the competition purpose we have to name them first, second, third. Only for that purpose the differentiation is done. So hope participants will take it very challengingly and very positively in such case. Yes or no? This is only for the competition purpose the gradings are being given. Otherwise, all participants, all posters, all presenters, all topics and themes are wonderful. So keeping in view of that, it was a very tight competition to the judges here to find out who's first, who is next, because I already told. So in order to encourage all the participants are going to get the cash awards as well as mementos along with certificates. Now, the first prize of the poster presentation of this 5-day workshop. Yes. Before going to announce the names, all the participants are very curious and eager to know. Before to that, I would like to invite Professor Pratiti Bhadra, our Pro-Chancellor Sir K.S. Jagannath Rao Garu, and convener of this 5-day workshop, the women who is behind of doing, able to do all these things on a single hand, Professor Vasuja Devi Madam, to give the cash award, certificate as well as mementos to the prize winners. I cordially invite Professor Subakar Raju sir to join with the guest. Sir, please sir. Please come on to the dais, sir. Of course, not on the dais, off the dais. And now, participants, you are curiously waiting, and definitely some of the participants are eagerly going to see on my sheet who is the list. So let me read out the names. The first prize goes to the top. Any guess? Yes. Any guesses?
You can guess yourself also.
The title, with the title, I would like to call upon the team, Gut Microbiome. With a big round of applause, Dr. Amrita and team. Can I call Dr. Amrita and team, ma'am? Arunita, Dr. Arunita and team. Gut microbiome. Yes. That is the true inspiration, right? When yesterday I was saying that I got inspiration from sir, you didn't believe. Now can you believe? Yes. Age is not at all a matter. Everything is here. We are no way old. We are all... Yes, age is only number. Yes, Pratiti ma'am. Thank you for helping me out. Age is only number. Everything is here. We are always saying as and when we are happy, smiling and positive. That's all. Yes, I request Pratiti ma'am to present to the winners, first prize winners, the cash prize of rupees 2,000. Best poster presentation award along with cash prize of rupees 2,000 and a memento. With a round of applause, join your hands together to encourage the first prize winners of today's post presentation. Congratulations. That's really wonderful. All the best. Wish you all the best. Congratulations. Thank you, sir. Any guess of second prize winners? Any guess? Okay, let me read out the title and then you can guess. TDP 43. Artik and team from KL University. Yes, all the team members. all the team members. And this team has won cash award of rupees 1500, best poster award. With a round of applause, join your hands together, dear participants. Congratulations, all the team members. And you know, when you observe the participants, keen observers, how many posters they prepared? 3, yes, 3 posters they prepared on TDP 43 and Trans Drive, yes. You can see, yes, they are going to showcase those posters now. Congratulations. Congratulations to all the team members. Congratulations. And I cordially invite Dr. Pratiti Vadra to felicitate. Yes. Cash award of rupees 1500 and a memento. Congratulations. And all the best to your students. Thank you. Thank you so much. Now, audience, you should not sleep. Come on, guess. You should not sleep. Any guess? Who is that Vishnu? Out of all this competition, I didn't find any poster presentation by Vishnu. But from the beginning, I am getting Vishnu. Okay, last present. Yes, your guess is right. But not now. Third prize winners. Title, let me read out the A and ML advances. Soumya and team. Yes. I invite Soumya and team to receive the cash prize of rupees 1000. With a big round of applause, join your hands together. They receive the best poster award along with cash prize of rupees 1000. Good. Congratulations. Soumya and team members, congratulations. Congratulations. Yes. Consolation, team wise I'll call as from the beginning Vishnu and team. That is Purusha and Prakriti title. Purusha and Prakriti title. Yes, consolation of cash award of rupees 500. Please come on to the dais. Yes. And then consolation awards goes to 4 teams. Aman and team. Yes, Aman and team. You are cordially invited to the dais to receive cash prize of rupees 500. Aman and team. Aman and team. Yes, along with your poster. Thank you. Congratulations. Congratulations, both of you. Aman and team, thank you. Next consolation award goes to Hypothesis Model for Treatment of Localized Brain Tumor, GBM, Nandini and team. They are awarded with rupees 500 cash award. Congratulations. Yes, your poster? Yes. I invite next cash awardee team, predicting mental integrity via EEG ML methods. A hypothesis testing. Yes. They have received cash award of rupees 500. Congratulations. Congratulations. Next consolation goes to the title, entitled with Purusha and Prakriti team. Cash award of rupees 500. Let's congratulate them with a round of applause. And dear participants, this is not the matter of this cash award. The best poster presenter awardees will be receiving the soft copy of best poster recipient award through certificate through their emails. They would be receiving through soft copy to your mails. You will be receiving the best poster awardee certificates. I congratulate all the poster presenters and I wish you all the best.
Vasuja?
Sir. Outside college.
Thanks to all of you for your wonderful participation. I should say, the woman behind this program is Vasuja Devi. She is the convener and she got DST SCRB award of 2 lakhs, for conducting this program. You can't believe, for last 1 to 1.5 months, she's not sleeping, honestly. Because she's also trying to apply many other grants for her own, upliftment. She's building up AML application brain. So she's going to be a part of many of the games. So I should really congratulate Dr. Vasuja Devi for a wonderful, sincere, dedicated, hardworking, whatnot. She has done the best job. I know she lost a lot of her time, with family and other thing, but she made it. Very, very proud of her, and let us give a big applause to her. I have 2 women who always, the third woman, girl is in exam. She's not around. She's my own student, Soumya. See, I have, Shobha Rani and Anita. See, you are carrying your bags, jute bags, It is a hard work of them because we have a women tech park and, they were working day and night. And suddenly we made a decision that we would like to have even bags for children yesterday. Imagine 250 children. They work till night 11 o'clock. So these two people, extraordinary job they have done. All the time writing emails, taking care of things, we can't imagine that our thanks is not simple for them, but they have done a splendid job. The other girl is in exam, so in absentia, I say congratulations to Soumya. This is a big team. This is a big team, and they work, like a process. We have another woman behind the success is Pavani, madam. See, this girl, the cute of this girl is always smiling. You never see her, any inch of sadness. I think she has a good alpha wave in her brain, all the time keep active and keep smiling, that inspire people. She took her time and did an extraordinary excellent job. And we have a lot of students coming from NCC, NSS, and also volunteers, all the students, please come here.
Pharmacy volunteers team,
Pharmacy, we have
CSC
biotechnology,
volunteers team,
all these people
ADS.
working day and night.
Please come
for
onto the desk. Everyone who have helped me, everyone.
Name by name, we cannot.
Please come onto the dais. ఇక్కడ నుంచి ఉండొచ్చు అందరూ. ప్లీజ్ కమ్ ఆన్ టు ద డయాస్. ఇక్కడికి నితిన్ ఎక్కాలి. విద్య, నందు, నూర్, లక్ష్మి.
Lakshmi is here.
నితిన్, గాయత్రి, తేజు, హర్ష, everyone.
We call them, these are our heroes. They are the people, behind the whole process. Even night 10 o'clock, 11 o'clock, chief guests are coming, receiving them, putting them in order, taking care of all the responsibility. Extraordinary, extraordinary, extraordinary job. We're really proud of this team. And now they become our team, so we keep using them for many activities. But they are not only working as volunteers, but they are learning a lot of things. But they're around, they keep listening to people, understand many things, and also they know how to organize. They met many people from outside, they know how to deal with the people. Because many of them are in the first year, second year of their undergraduation. Extraordinary job they have done. We really congratulate them. And then we have NSS team, but NSS officer is not there. Subakar Raju, he's here, and his team, Srikanth and other thing, please sir. So these team helped us.
NSS students, please, anybody there, please come. NSS
students.
Anybody there, please come.
See, yesterday is a Herculean task, bringing 250 children, all maintaining their silence, keeping them in the bus, make sure they don't miss anywhere, organizing food, organizing everything. For them is an excellent job NCC, NSS team has done. So that's where we are really congratulating Subakar Raju. Of course, Srikanth is not here. Chandrasekhar is not there. These are the major people who worked on this process. The main important, you all enjoyed the food. Today was a feast because there was a bhaji, Mirchi bhaji, my special. I should congratulate Dr. Hema Malini, who is behind brain in making such a nice food for all of us. Excellent, excellent job. And then all the snacks we are getting, all supplied by, Satish, our canteen person. He's excellent. All the time you just tell him, he's available. Anything we would like to miss, we don't miss it. And at the end of this program, we're all going to have ice cream. So a different change, for all of you, from a tea to ice cream, we promoted, And I should thank our chairman, Koner Satyanarayana Garu, for all his support, what he was doing for this process. We also thank our Vice Chancellor, our Pro Vice Chancellors, Registrar, Deans, all these teams who are making the team. Important is this room is very crucial for us. See, all these people who are supporting, taking care of your, photograph. By evening, we'll have a photograph from them. I should really congratulate both of them, because by the time I go home by 6:37, I have the photograph. So I keep sending them to the speakers, everybody. So you maintain a very clear communication. I should congratulate all the team, technical team of this room, making these things happen. And we also have drivers who did a good job taking all of you safely to Guntur, bring you back. And our special thanks yesterday we communicated to Dr. Srinivas Reddy and his wife, who did an excellent job, hospital, showcasing the things, and also Dr. Alex from AIMS. So now we building up a networking. That's where we are doing, and then this team will keep continuing serving the student community, giving the new ideas, whatever the experiences we have gained, we would like to give you. Not only that, we will support no matter where you belong, not necessarily KL. You can belong to any institution. Please feel free to contact us. We help you all. Okay? That's the main theme. Let us make discoveries in India. That's where we are looking for. Okay, we always say this is discovered in Germany, this is discovered in Max Planck, this is discovered in Harvard, this is discovered in MIT. So a day should come that we are doing that kind of things, most challenging issues. I am very positive that the poster winners definitely will make 2 to 3 best quality, hypothesis reviews. And I am overwhelmed. I am very happy because I have a lot of energy. I could use it. So I'm very happy and healthy. In the next 10 years is going to be my healthy life because I enjoyed great science. Lot of new things I learned. A lot of collaborations we brought. And I should thank all of you for a wonderful job traveling all the way from to Vijayawada. Whatever we are doing, you enjoyed. We know there will be some mistakes, shortcomings. If anything, please excuse us. We are responsible. We are responsible for that. We'll try to correct them in next time, when we have the meetings. Thank you very much to all of you.
Dear participants, we know that the participants from out of Vijayawada are eagerly waiting because the time schedule of trains or flights or buses are as it is after 3:30. Many of them have informed to the convener of the workshop. Keeping in view of that, we are calling to distribute the certificates in prior hand out of AP, out of Vijayawada city people. And KL students, I hope you understand, because the need of the hour is the participants, as they have to report to their organizations by tomorrow after doing a long journey. So we are going to call the participants who are from out of Vijayawada first, and I request now the convener of this workshop, Professor Vasuja Devi Madam, Associate Dean, Student Affairs, Vasudha Devi Madam, onto the dais to distribute the certificates to the participants, along with our beloved Pro-Chancellor, Sir Jagannath Rao, Sir, along with Professor Pratiti Badra Madam. Please. Yes. Dear participants, when Sir was saying that among the team members, the crucial role played by two main members, Professor Vasudha Devi Madam and Soumya is back from the exam. Today she has completed her pre-PhD written exam. She is Soumya. Sir was talking about Soumya, who is working under him. So we really feel jealous of seeing her because she is under the guidance of our beloved Pro-Chancellor, Sir. So she has completed her internships in abroad. One is in Japan, if I'm not wrong. One is in Japan for 3 months, and another one is in for Japan in 6 months. Another is in
Conference
another one conference she has attended with the best conference speaker award in USA under the guidance of our Pro Chancellor Sir Jagannath Rao, sir. And she is going to have a collaboration with international collaborators like Hegde and one more, Samuel Abraham, sir. So very soon we are going to see her in abroad with her collaborative work, and let us wish all the best to her. Now I request our yes. Now I request our KL Authority delegates, our Pro Chancellor Sir Jagannath Rao Sir, Vasuja Devi Madam, Pratiti Ma'am, D. Srinivas Sir from the Department of ECE, please Sir, and Subhakar Raju Sir. Kindly give the certificates to the participants. Please Sir. Sir, Srinivas Sir. Atiro M. Das from AIMS Raipur. I would like to call upon Arunita. I would like to call upon Siddhartha. Arunita. Yes, ma'am. Siddhartha. Yeah. Siddhartha. Siddhartha. Yes. I would like to call upon Lalita Sahitya. Oh, KLU alumni. Congratulations. Oh. Great. I would like to call upon Saumya, Sai Saumya. Thank you. Next, Anusha. I would like to call upon Anusha. I would like to call upon Muhammad Nazia. And I would like to request T. Uma from Sri Venkateshwara Medical College. T. Uma garu. And I would like to call upon Nirmala Madhuri garu.
Stand here, right? Stand here.
Nirmala Madhuri. G. Madhuri. I would like to call upon G. Madhuri garu.
Any outsider people are there, like non-KL University people, please come. If anybody missed to take the certificate and all, please come. Outside participants